Cool. All right. Hi, everyone. I'm going to share screen. Hi, everyone. My name's Ethan. I'm here with Elise, who's a volunteer from UBS, and we'll be running the bio lecture today. So it's going from 1:30 to 4:30. I'll show you a bit of a rundown of the game plan for today. But first, I'd like to acknowledge the traditional custodians of the land we're gathered on today. Pay my respects to the elders, past and present, for they hold the memories, the traditions, the culture, and hopes of the Aboriginal and Torres Strait Islander peoples across the nation. All right, so this is the game plan. We're just going to be splitting into halves. So 1:30 to 3 o'clock, we'll be doing Unit 3, Area Study 1 and 2. Then we'll have a break, and we're actually bringing in a VC examiner for a short Q&A. And then we'll continue with 3.2 and try and get Unit 4 done as well. We only have 3 hours, so I won't be able to cover everything, and I'll be going pretty quickly. But don't stress too much if you miss something. There will be a recording, which we'll distribute at the end. There are also 4 mini pop quizzes, which have nothing to do with bio, and that's just to make sure everyone's awake and hopefully have a little bit of fun. All right, and I might skip a few practice questions if we're running short on time. All right, so this is Elise. Elise, do you want to give a bit of an intro?
Yes, hi everyone. I'm Elise. I work for UBS, who have partnered with Skyline for this program. A little bit about myself. I finished year 12 in 2015, so 5 years ago now. Just crazy how much time's passed. I actually did the International Baccalaureate. I didn't do VCE, so my experience is a little bit different to what you guys are going through, but very similar in terms of the stress of year 12. I was one of my 6 subjects. I enjoyed it. It was good. It was a long time ago, so I don't remember as much as I'm sure Ethan can teach you. I then did a commerce degree, and now I'm working at UBS. This is my first year, and I'm working in the metals and mining team. Yeah, if you have any questions today, just feel free to message me privately, and then I can speak up to Ethan and get your questions answered.
No worries. Thanks, Elise. My name's Ethan. So far in lockdown, I've been playing Among Us a lot. I have really bad handwriting, so good luck reading that today. And I've been currently obsessed with The Umbrella Academy and The Boys. Watch them almost every night. All right, so we'll start off with a quick pop quiz. If you could get the poll up, Andrew. Name this album. And then we'll probably give it like 20 seconds. All right, so this was actually Parachutes by Coldplay. Can we see the results, Andrew, please? Awesome, cool. So most people got Parachutes, definitely not Foo Fighters. Anyway, cool. So this is, if you've ever used Poll Everywhere, this is kind of like that. It's just so you can ask me a few questions. So if you've got an iPhone, I think you can use the camera app and scan that QR code. Otherwise, go to menti.com and type in that code, and that's just going to be in the top right of every screen. So you can ask me questions, which I'll address halfway through and then at the end as well. And also feel free to message the chat or at least any questions you have. Okay, so if people don't know, the biology study design should be your guide for how you study biology. What I did is I went to look up biology study design, go to the second link. I looked at that document, and they can ask you anything on there, and they can't ask you anything that's not on there. So that's literally what is going to be on the exam, the biology study design. It looks like that. And what I did was I printed it off and I highlighted all the stuff that I didn't know, and then I kind of ticked off the stuff that I did know, and that way I made sure that I knew everything on the course. All right, so this is the first dot point on the study design. So it's the first thing we need to know about. We need to know about the plasma membrane. And the thing is, with biology, there's a few, whenever you're going through a definition, there are certain keywords that you have to have in every time. So with the plasma membrane, you want to know that it's an active boundary. And what that means is that it controls the movement of substances into and out of the cell. You also want to know that it's described by the fluid mosaic model, and that's kind of like a theory of how it works. So it means that it's a two-dimensional fluid, which is the phospholipid bilayer, and it's got associated embedded proteins, and that's what makes the mosaic out of it. So it's phospholipid bilayer and associated proteins. And if you have all those keywords, you should be able to get full marks for that question. And I will be distributing my notes hopefully, and that'll have all your definitions in it. So this is a plasma membrane. Each of these is a phospholipid. This is a cholesterol molecule. With the phospholipids, you've got a hydrophilic phosphate head. So hydrophilic means it likes water, and that's why the phosphate head will face the outside and the inside of the cell. So they like water. The lipid tails do not like water, so they're hydrophobic. That's why they face each other. Then you also have cholesterol, which is just a molecule that's embedded in the plasma membrane, and it keeps it all fluid at really low temperatures, and then it keeps it together at high temperatures, and that, again, that's in my notes as well. With your integral proteins, that's a protein that spans both layers of the membrane, and your peripheral proteins are just on one layer or they're associated with one layer. We also need to know about how stuff moves across the plasma membrane. So if you've got a high concentration of molecules here and a low concentration of molecules here, your molecules are going to want to come into the cell. So they start pushing downwards because they want to spread out. If that molecule is hydrophobic, it can pass these lipid tails. So it can go straight through the plasma membrane. So it's going to go through path A. If your molecule is hydrophilic, it likes water, so it can't pass through the lipid tails. It's going to go through the protein channel. And if you want to move your molecule in the opposite direction to what it's pushing, so if you go against the concentration gradient, you're going to have to use a carrier protein or a protein pump. And that's going to be via active transport. So simple diffusion is when it diffuses down a concentration gradient directly across the phospholipid bilayer. Facilitated diffusion is when it diffuses through a protein channel or carrier protein. Active transport is when it's pumped out against the concentration gradient using a protein pump or carrier protein, and that uses up ATP. Then with osmosis, that's specifically the diffusion of water, and that goes from low solute concentration, so here, to high solute concentration, so outside. So it goes from hypotonic area to a hypertonic area. All right. So have a go at this question, and then we'll go through it. Right, I might just give this one like 20 seconds or so. Alright, so most people chose C, which is good because that is the answer. Whoops. So with A, large molecules pass the plasma membrane by simple diffusion through protein channels. Simple diffusion is directly through the phospholipid bilayer, not protein channels. Small charged ions pass the plasma membrane by facilitated diffusion. Yeah, that's good. Directly through phospholipid bilayer, no. So facilitated diffusion is through protein channels or carrier proteins. Very large substances enter the cell by endocytosis, so that's things like entire cells. They'll be phagocytosed, so that's all good. And an important thing to note is that that requires ATP. Water passes the plasma membrane by facilitated diffusion through carrier proteins. No, so water passes the plasma membrane through osmosis, and if it does go through by facilitated diffusion, for example in the kidneys, I think that's a very specific spot where it happens. That's through aquaporins, which are a type of protein channel, not a carrier protein. All right, so nucleic acids. We've got a double strand of DNA here. What we need to know is that the 5' end, you don't need to know about the chemistry of it, but the 5' end is the end with the phosphate. The 3' end is the end with the sugar. So a nucleotide is made up of phosphate, sugar, and a base. In DNA, you've got A, T, G, and C. In uracil, you've got A, U, G, and C. And you'll notice that the 5' end matches up with the 3' end of the opposite strand. So that means they run anti-parallel. Another thing to note is that there are hydrogen bonds keeping them together, keeping the two strands together, and between the same strand, they're combined via covalent bonds, which are a lot stronger than those hydrogen bonds. All right, and one nucleotide is the monomer of the DNA, and that's made up of phosphate, sugar, and a base. All right, so this is, you need to know about how to compare DNA and RNA. Basically, DNA is double-stranded, RNA is single-stranded. There are some cases where RNA can be double-stranded, but that's very rare and usually in viruses. So we can say double versus single in a VCAA context. The subunit for DNA is deoxyribose sugar, and for RNA it's ribose sugar. And the base is thymine in DNA and uracil in RNA. So this is your codon table. This is basically your dictionary for how to make proteins. So your mRNA comes from the nucleus after transcription, and we'll go through this as well. And your ribosome reads the mRNA, and then it looks to this dictionary to try and figure out what each of the codons mean. So UUU, that tells the ribosome to place a PHE amino acid. So the codon here is your mRNA. The letters here are your amino acid, and those are brought to the ribosome by the tRNA. There are also stop codons, which tell us to stop translation, and a start codon here, AUG. So the reason that DNA has lots of codons which code for the same amino acid. So you can see all 6 of these code for LEU. And that means that the code is redundant, and another word for that is degenerate. And then another property of DNA that you have to know is that it's universal, which means that you can stick DNA in any organism and it'll work. It'll produce the same protein. So with gene expression, start off in the nucleus with DNA. We transcribe it to mRNA using RNA polymerase and all that stuff. Then the mRNA moves to the ribosome. So if you want to think of it like copying down a recipe, you go to your grandma's house, open up a cookbook, you write down some notes in your notepad about how to make a cake, for example. That's your mRNA. Then you take it to your kitchen, that's the ribosome, and you make the protein, which is the cake. So transcription. You do need to know all the steps involved, and that's put in detail in my notes. So RNA polymerase attaches to a promoter sequence, and that's just the start of your gene. So it unwinds the DNA, and it starts placing RNA nucleotides that are complementary to the DNA template strand. It places them in a 5' to 3' direction. Now that's a really important thing to remember. When you're placing nucleotides, they always have to go from 5' to 3', which means if you go along the template strand, it's actually 3' to 5'. So they're running anti-parallel. You also get a mark for saying uracil is used instead of thymine, and then once we produce that pre-mRNA, it detaches and we undergo post-transcription modification. So that involves adding a poly-A tail to the 3' end, a methyl cap to the 5' end, and then removing introns and sticking the exons together. So the introns don't really tell us anything, they're pretty useless, but your exons are useful, so we stick those together. And then we have a mRNA strand. So you've got your poly-A tail, and then you've got your methyl cap, and that's your final mRNA. So the mRNA moves to the ribosome, where it undergoes translation. So the ribosome attaches to the mRNA strand, and it starts reading it in groups of 3 called codons. So each of these is a codon, and we'll start off with the start codon, which is always AUG. And the tRNA comes along and it brings an amino acid. Now it's really important that we remember that it's a specific amino acid. So the tRNA has an anticodon here, and that anticodon, if we look at the codon table, codes for one specific amino acid. It doesn't attach to any other amino acid, so ACC will always bind to TRP. So it brings that anticodon, it brings the amino acid to the ribosome, it bonds here via hydrogen bonds, and the amino acids join up via condensation reaction, condensation polymerization, and then eventually we get a polypeptide when we get lots of amino acids, and when we reach the stop codon, that's when we finish. So you have to label how we're starting, how we're continuing, and then how we finish as well for full marks. So types of RNA. We mentioned messenger RNA, so that's created in transcription. We've mentioned transfer RNA, so that brings a specific amino acid to the ribosome. And then ribosomal RNA just forms the structure of the ribosome. So it's all the stuff in here. That's rRNA. Cool. So a little bit about organelles. I was going to get you guys to guess what all these are, but I don't think we'll have that much time. So this is a mitochondrion. Hopefully we all know that one. This is smooth ER, and this is rough ER. And the main difference between those is you can see these red dots. Those are ribosomes, which make it rough. D is the nucleolus, and that's where we're creating rRNA. It's for the ribosomes. E is the nucleus, that's where we're storing our chromosomes. F is the cytoskeleton, that holds the structure of the cell. G is free-floating ribosomes, and the difference between the free-floating ribosomes and the rough ER is that the free-floating ribosomes produces proteins that stay in the cell. With the rough ER, those proteins are exported, and that's a really important thing to remember. H is our Golgi, and that packages and modifies things for export from the cell. Alright, so those are the answers. Cool. So, steps in the manufacture and export of proteins. So, if a protein is destined to be exported from the cell, so if we're going to chuck it out of the cell, they're going to be made in the ribosome. They're going to be folded in the rough ER. Then they move to the Golgi via transition vesicles. Once in the Golgi, they may be modified, so they may get some groups attached to it, which make it more functional, so for perhaps a carbohydrate group. And then once in the Golgi, they're made into secretory vesicles, which fuse with the membrane and expel the contents via exocytosis. So, in terms of the diversity of proteins, you don't need to remember all this stuff. I think it's popped up once ever in biology, where you just had to name one type of protein and then give an example. But it shouldn't be too hard. You can take a screenshot if you want, but it's also in my notes, so don't worry too much. Rich is just the mnemonic. Basically, you just need to know that proteins have lots of different functions. So the proteome is the total proteins produced by a functioning organism or cell, and the reason that we study it is because proteins interact with each other, and some are reliant on others. So there's no point studying proteins individually, because that won't tell us a lot about their function. To know their function, we have to have a look at how they interact with other proteins. Alright, so structure of an amino acid. We do need to know this as well. This is important content. So we've got a carbon in the middle, then we've got an amine group here, a carboxyl group here, and an R variable side chain. So it's a variable group or R group or side chain, you can call it any of those. Basically what the R group does is it gives the amino acid certain properties. So the R group can be anything. There's 20 different things that it can be. And that R group determines how the amino acid interacts with other amino acids. And that's going to determine how your protein will shape in the end, or how it'll look in the end. Alright, the peptide bond is just a bond, the covalent bond between two different amino acids. So that's that bond over there. And it forms via a condensation polymerization reaction. So condensation means we're squishing stuff together, and you might also remember it because a water molecule is released. And then if we're going backwards, we need to use up a water molecule, so we call it a hydrolysis reaction. Hydro means water, lysis means breaking up. So instead of producing a water when we're going backwards, we're going to be breaking up a water to get two different amino acids. So with the 4 levels of structure, this is something that a lot of people kind of miss a few keywords on it, so it's important to pay attention to this one. So the primary structure is the specific linear sequence of amino acids, and that is the definition you'll have to use on your exam. So it's the sequence of amino acids, and it's a linear sequence. So we're not describing the interactions between amino acids in tertiary structure or via like folding and all that. We're just looking at how they are ordered at the moment. And they're joined together by peptide bonds, which are very strong covalent bonds. With your secondary structure, this is localized coiling and folding into alpha chains and beta pleated sheets, alpha helixes and beta pleated sheets. And that's via hydrogen bonds. So you do need to know what kind of bonds hold the secondary structure together. So that's these two things. With the tertiary structure, it's a little bit different because instead of being between the carboxyl group and the amine group, it's actually between R groups in the tertiary structure. And that's how we get the overall three-dimensional shape of a protein. And it's more globalized folding. So it's joining amino acids that are very far from each other on the chain. So remember, tertiary structure is global, secondary structure is local. Tertiary structure is between R groups, secondary structure is between carboxyl and amine groups. Then we also need to know what kind of bonds are in tertiary structure, that's in my notes as well. With quaternary structure, that's the arrangement of two or more polypeptide chains, and that just forms the final protein. Not every protein needs a quaternary structure. So have a go at this question. I might just give 30 seconds for all of them. Also, let me know if you guys think I'm going too quickly. I think we are going well for time, so I can slow down a bit. All right, so most people chose A, which is correct. So a change in the tertiary structure of a protein may result in the protein becoming biologically inactive. So a protein becomes biologically inactive when its shape is changed. So the tertiary structure determines the overall three-dimensional shape of a protein, and if the tertiary structure breaks down, the shape doesn't have the proper shape. This protein doesn't have the proper shape, and therefore the function is lost. Proteins with a quaternary structure will be more active. No, because not all proteins have quaternary structure, and quaternary structure doesn't really tell us a lot about how active it is. The shape tells us about the activity. Two different proteins with the same number of amino acids won't be the same number. If it has the same amino acids in the same order, then it'll have identical functions. The number of amino acids doesn't really come into it. Denaturation will alter the primary structure. No, primary structure is very strong because it's combined via covalent bonds. All right, so gene structure. We need to know about structural genes and regulatory genes. Structural genes are genes that code for the structure and functioning of an organism. So anything pretty much except for regulatory genes. Now, regulatory genes control how often structural genes are expressed. So regulatory genes can produce some repressor, which binds to the operator here, and it switches off the structural gene. They can also switch on a structural gene. So they might be enhancing, or they might be repressing the structural gene. And this is via transcription factors. All right, so just quickly with prokaryotic genes, the entire transcribed mRNA is translated. So there's no introns with prokaryotes because they don't have a nucleus, therefore they can't undergo post-transcription modification. And as you can see with my crappy diagram here, the mRNA can be produced via transcription and it can be used in translation at the exact same time. With alternative splicing, this is how we can get different proteins from the same from the same gene. So in different cells, if you need different proteins from the same gene, you might get a different arrangement of exons, which will give you different proteins. So we can keep some exons and take out some exons, and that's all up to the cell. So if the cell wants certain exons, it can keep it and it produces a different protein. And I think you can also keep some introns as well. With the lac operon, this is a specific operon that we need to know about. So basically with some prokaryotes, they only want to produce this gene that breaks down lactose if lactose is present. So if lactose is not present, we're not going to be producing lactase, which is the enzyme that breaks down lactose, because that's just a waste of energy and resources. So what they do is they use this regulatory protein, which produces a repressor molecule. So this repressor molecule, if lactose is not present, it'll come and bind to the operator. So it says, hold on, RNA polymerase, I don't want you transcribing this gene because you're not needed. So it'll switch off the gene. So the gene is switched off when lactose is not present. When lactose is present, it is converted to allolactose, and so you can call it allolactose. Allolactose is pretty much the same thing. And then allolactose will bind to your repressor, and that'll bring the repressor away from the operator. So now the gene is switched on. It's like a gate. So the gate is now open. RNA polymerase can bind to the promoter and transcribe the gene. So what happens is lactose will bind to the repressor, and the repressor will change shape, and that's a really important keyword to have in your answer. So it changes shape and detaches from the operator. Okay, so have a go at this question. All right. So the answer was B. Could we just see the results, please? Okay, cool. So yeah, most people chose B, which is good. The RNA polymerase will bind to the promoter region. Or the operator region is where the repressor protein binds, not the RNA polymerase. The regulatory gene is what produces the repressor. And the structural genes are just your lactase, enzyme genes, and other stuff. There's another question here. Have a go at this one. I'm just going to close my window. Okay, can you let me know if that's better, Gypsy? Okay, awesome. All right, so most people chose C, which is good. So the repressor molecule will bind to the allolactose. So I'll go back to that diagram here. The repressor binds to the allolactose and it detaches from the operator. If the structural genes in the lac operon are being transcribed, remember that means that the operon is on. So we are going to be producing that protein. Which means that A is not correct because the operon will be on. Allolactose binds to the promoter. No, the RNA polymerase binds to the promoter. Regulatory genes produce RNA polymerase. RNA polymerase is not produced by regulatory genes. Regulatory genes produce the repressor protein. All right, so having a look at enzymes. Your keywords here are they are protein catalysts. They increase the rate of specific biochemical reactions, and they do that by decreasing the activation necessary for that reaction to occur. And they do that by providing an alternative reaction pathway. And an important thing to note is that they don't get permanently changed by helping out this reaction. So that's just being shown in this diagram here. So your substrate will be perfectly complementary to the not exactly perfectly, but it will be complementary to the active site of the enzyme, it'll bind, it'll react, and then the enzyme returns to its normal shape. And that shape is determined by its tertiary structure. Cofactors are little things that speed up or enhance the action of an enzyme. So enzymes are enhancing the reactions, they're speeding up reactions. Cofactors help enzymes do that. So they enhance the action of an enzyme by binding to the enzyme and changing the shape of its active site. So it's making the active site even more complementary to its substrate. So they can bind to the active site or any other site. Most act as carrier molecules, so that's things like ATP, NADPH, NADH, and we'll cover those in respiration and photosynthesis. And they can be organic, so these ones are organic coenzymes. Your inorganic cofactors are metal ions. And another thing to note is that organic coenzymes are not proteins, but they are organic. So your types of reactions, you've got anabolic and catabolic. To remember this, you can kind of remember like people taking anabolic steroids to get bigger, to get more muscles. Anabolic means you're putting stuff together. So you're building muscle with anabolic steroids. And they require an energy input. So think of it as sticking two pieces of Lego together. You need to push in energy to make them, to put two things together. Then when you're breaking stuff apart, you have to put in a little bit of energy. So that's your activation energy. And then once they fly it apart, that's releasing a lot of energy. So that's going to be exergonic. So these are the factors that affect the activity of enzymes. So we've got substrate concentration here. If we get more substrate, there's going to be more collisions between substrate and enzyme. And that means your rate's going to increase until you reach this point, and that's called the saturation point. So the saturation point is where all your enzymes are being used up at once. So we're not going to get any faster after that point because there's not enough enzymes to increase the rate of reaction. They're all being used up at once. With temperature, the rate of reaction will increase because there's more energy in the mixture, there's more interactions between particles. Until you get it to a certain point, that's the point of denaturation, where your tertiary structure is going to fall apart because there's so much energy that the bonds holding tertiary structure together break apart, and your protein, your enzyme, is not going to have the right shape anymore. So that's when the rate of reaction plummets. And then we can't go backwards from that. Then the enzyme is finished. With an increase in enzyme concentration, there's just an increase in the rate of reaction. There's no plateau because you can't actually saturate a substrate. You can only saturate enzymes. With pH, pH determines the shape of your active site just because of the different ions in your solution. So pH, don't have to worry too much about the chemistry of it, but pH is how much hydrogen ions you have and how much hydroxide ions you have. And the ratio of those ions determines how your enzyme will look. So it determines the interactions between tertiary structure. So at the optimal pH, that's when you have a perfect shape. And when you go further away, it's going to have not as good shape. And that's just an explanation of those concepts, but it is a little bit of a lower yield. So don't worry too much about it. It is in my notes. With inhibition, obviously you got irreversible and reversible, is kind of self-explanatory. So with irreversible, they don't detach from the enzyme. So an inhibitor attaches to the enzyme, it stops it from working. Irreversible inhibition is when it binds covalently. So remember, those covalent bonds are very strong. It will not detach. With reversible inhibition is usually to the active site and it bonds non-covalently. With competitive inhibition, that's when it binds to the active site. Non-competitive inhibition is when it binds to the allosteric site. So allo means different. The allosteric site, when it binds to the allosteric site, it will cause a change of shape of the enzyme, and that's what causes the active site shape to change so that it's no longer able to bind. So in both cases, we both have to mention that it's no longer able to bind to a substrate. So the lock and key model and the induced fit are two models. So again, models means like different theories. The lock and key model says that the active site is perfectly complementary to its substrate. Therefore, it binds, it doesn't change shape at all, and then it unbinds. With the induced fit model, it says that the active site is relatively complementary to the substrate, but then it also changes shape a little bit. So you can see it's not perfectly complementary to the substrate here. It changes shape, and then once it's finished interacting, it goes back to its original shape. And that just allows for different molecules to use the same enzyme. So slightly different shapes can use the same enzyme. We'll have a go at this question now. All right, so most people chose B, and that was the correct answer. So phosphofructokinase, you can kind of guess what it is by the suffix. Enzymes will usually end in ase. So phosphofructokinase is an enzyme, and enzymes speed up the rate of reaction. A coenzyme is this ATP. So ATP is donating energy to the reaction, and so it's carrying energy from another source to the reaction, and that acts as the coenzyme. The substrate for the reaction is fructose, and the product would be this fructose, and then it releases energy in the form of ATP. ATP will release energy and become ADP, but ADP itself does not hold that much energy. The form that holds energy is ATP, so it's not releasing energy. This reaction is actually using up the energy from the ATP. Let's have a go at this question now. All right, good. C. So, what happens here is that we've got two test tubes at different temperatures, and the trick here, it is a little bit of a dodgy question because it doesn't tell you if the reaction is reaching completion. If the reaction did reach completion, then I think some of the other answers might have been more correct. But in this case, they were assuming that the reaction did not reach completion. So, at a lower temperature, there will be fewer collisions between substrate and enzyme, and that's going to mean there will be a lower amount of glucose produced in the end. The enzyme's active site would have denatured. They would not have denatured at low temperatures. They only denature at high temperatures. Lowering the temperature does affect the digestion of lactose, just in how fast it is. It doesn't stop the digestion, it just makes it a bit slower. Two test tubes contain the same amount of lactose and lactase enzyme. I think that would have been correct if the reaction had reached completion, but I think they're making the suggestion that it didn't reach completion. All right. So with photosynthesis, we need to know about its purpose and the stages involved and a bit about their bacterial origins. So with photosynthesis, it's a series of reactions. So in your definition, you want to say it's a series of reactions. Remember, with your definitions, you want to say what it is. Don't just describe what it does. What it is a series of reactions. So it takes light energy from the sun, it makes inorganic molecules, combines them to form glucose. So your reaction equation will be 6 CO2 plus 6 H2O in the presence of sunlight and chlorophyll gives you glucose and oxygen. Some people like to put 12 H2O and plus 6 H2O here, but I think in the interest of balancing equations, I prefer this equation. You can do this one, that's perfectly fine, and I'll explain why that works in the end. So, we need to know a bit about the structure of the chloroplast. Have a read through a few of these and have a think about your answers, and then I'll go through the answers as well. So I'll just give you like 15 seconds to have a skim through. Oh, right, so one is the outer membrane, two is going to be the intermembrane space, and that's because it's between the outer membrane and the inner membrane. And as we'll go through later, the outer membrane can be considered to be part of the host cell, so the eukaryotic cell's membrane. The inner membrane is more related to the chloroplast, and we'll kind of go through that as well. 4 is kind of the cytosol of the chloroplast, we call it the stroma. 5 is thylakoid lumen, so lumen means the space inside. 6 is the thylakoid membrane. 8 is a thylakoid or lamella. Lamella more specifically refers to the things that go between stacks of thylakoids. The stacks are called grana. Don't worry too much about the purple and yellow ones, those are a bit harder. So the purple one is starch, the yellow one is a lipid droplet. This one you should know, this is DNA in a circular chromosome. The red things or pink things, those are ribosomes. And chloroplasts have their own ribosomes. So photosynthesis, this is what happens. In your light-dependent stage, that is the one on the left, you get water coming into the chloroplast, and light comes in as well. Light is trapped by the chlorophyll. And the chlorophyll uses the light energy to break water apart. So water is broken up into hydrogen ions, and those get carried away by NADPH and the electrons as well, and oxygen ions, which combine to form O2. And that O2 is a byproduct, so it's released. ATP also comes along. It's produced. ADP plus PI comes along, picks up energy, and then it becomes ATP. Then ATP and NADPH cycle back to the Calvin cycle. This is the light independent stage. And they use that energy to combine CO2 molecules together to make glucose, your sugar. You can also produce starch from this. And then those molecules, once they've unloaded their energy, hydrogen ions and electrons, they cycle back to the light dependent stage. So even though this is the light independent stage, it's dependent on these carrier molecules, and those carrier molecules are only produced when we have light. So light independent stage is still dependent on light, even though it's not directly so. And that might be a little bit confusing, but we will do a few bit of discussion on that as well. So there are some factors that affect the rate of photosynthesis, and we need to know these. So with light intensity, it kind of looks like the substrate concentration curve. So it'll increase up to a certain point. And that point is when there are limiting factors keeping the rate of reaction down. So limiting factors might be concentration of CO2, it might be amount of chlorophyll present, might be amount of chloroplasts. All these things contribute to, well, if they weren't high, if you don't have enough CO2, your rate of reaction is going to stay constant. With your wavelength, your light wavelength kind of determines how much energy is being absorbed by a chlorophyll. So if you've got green chlorophyll, it's going to be absorbing a lot of blue and red light, and that's why it's reflecting green. So green isn't being absorbed, so it gets reflected. If you had blue pigment instead of chlorophyll, it would be absorbing a lot of red and green light, but not a lot of blue light. So it'll reflect the blue light. And that means it'll get most of its energy from red and green. with your temperature, kind of looks like the enzyme curve. So it'll come up and then come down once the enzymes denature. With your CO2 concentration, that looks like the substrate curve, so that it reaches the saturation point. And at that point, there are other limiting factors, such as enzyme concentration, amount of chlorophyll, etc., etc. Now we have to know a bit about the same thing with aerobic respiration. So aerobic respiration is when we're using oxygen. Cellular respiration is any kind of respiration where we're getting energy from organic compounds. Aerobic is specifically when we're using oxygen. So we're oxidizing organic compounds and releasing their energy in the form of ATP. Some bacteria can respire aerobically despite not having mitochondria. That's an important thing to remember. So some bacteria don't need mitochondria, but we definitely do. All right, so have a think about what each of these are, and then we'll discuss them in like 15 seconds, 15, 20 seconds. All right, so the intermembrane space is going to be the space between the inner membrane, which is here, and the outer membrane, which is here. So inter means between. Your cristae are the folds of your inner membrane. So when the inner membrane folds, we call that a cristae. Ribosomes are these red dots, the black dots in this case. Your chromosomes made of the squiggly stuff. And the matrix is like the cytosol of your mitochondria. Important not to confuse the matrix with the stroma, and the stroma is for the chloroplast. All right, so this is a quick summary of aerobic respiration. Glycolysis happens in the cytosol, and we get 2 ATP. We have to remember how much ATP we get at each stage. So glycolysis converts glucose to pyruvate. Pyruvate enters the mitochondria, and then undergoes a linking reaction, which converts the pyruvate to acetyl-CoA. Then acetyl-CoA enters the Krebs cycle, produces 2 ATP and a lot of electron carriers. So it also produces a lot of NADH, and I'll go through specific amounts. And it also produces FADH2, which go to the electron transport chain, and they're used to produce 32 to 34 ATP. And it is up for debate how much ATP is actually produced, but in terms of theoretical amounts, it's 32 to 34. Some textbooks say 26 because that's more realistic, but I think in terms of VCAA, they want you to say 32 to 34. Glycolysis is just a breakdown of glucose into 2-pyruvate. Don't worry about these chemical structures. We also get 2 ATP from that, as I mentioned, and then we also get 2 NADH, which will cycle back to your electron transport chain. The Krebs cycle, this is the linking reaction at the start, and sometimes we consider that part of the Krebs cycle. The linking reaction pretty much just converts the pyruvate into acetyl-CoA so that it can enter the Krebs cycle. Pyruvate itself can't enter the Krebs cycle. The Krebs cycle happens in the matrix, and through the linking reaction, so this is a linking reaction, through the linking reaction we get 2 CO2 per 2 pyruvate, we get 2 NADH. Then through the actual Krebs cycle, we get 4 CO2, 6 NADH, and 2 FADH2. Then we also get 2 ATP. And a better way of remembering that with the CO2s is because glucose starts off as a 6-carbon molecule. So there's 6 carbons in each molecule of glucose. Acetyl-CoA is a 2-carbon molecule, and pyruvate is a 3-carbon molecule. So we can see here that per pyruvate, we're actually losing 1 carbon. And that's going to come off as CO2. And then the rest of the carbons, so there'll be 2 acetyl-CoAs here, which means there's 4 carbons that need to come off, and so that'll come off as 4 CO2s. An important thing to remember here, which I got confused with when I was studying bio, is that O2 is not an input to the stage. The O2 and the CO2 just comes from the glucose. It doesn't come from oxygen, free-floating oxygen. The electron transport chain is a bit harder to understand, so don't worry, or you do need to know it, but don't stress about it. So what happens is your NADH and FADH2 comes to this cytochrome here, these electron transport carriers. You don't need to know the names of them, so don't stress about that. You just need to know that they donate an electron, and those electrons are carried between these proteins here in the inner membrane. Inner membrane. And as they're carried along, they release energy. And then they use that energy, these protein carriers use that energy to pump hydrogen ions into the intermembrane space. Yeah, I don't know why that's erasing. That's the intermembrane space. So it's going against the concentration gradient, so there's a very high concentration of hydrogen ions in this space. So it needs to use that energy from the electrons. And now since we've got so much hydrogen ions in this intermembrane space, they're going to want to come back down. So they come back down via ATP synthase. You do need to know that protein. And ATP synthase is kind of like a windmill. It takes the power, like the wind of the hydrogen ions coming through, and it uses that energy to put ADP and PY together, and that produces ATP. So we're using that energy to produce ATP, and that produces 32 to 34 ATP. And the hydrogen ions come here, they combine with some oxygen, so there's oxygen coming in as well, combined with the hydrogen ions and the electrons to produce water. So oxygen is known as the final electron acceptor because it accepts the electrons after this whole chain, and we produce water at the end of this stage. So this is a quick table summarizing all of that. Feel free to screenshot it. It is also in my notes, but I'll give you a few seconds to screenshot if you want. All right, so anaerobic respiration. This happens when O2 is toxic to bacteria. So some bacteria actually can't deal with O2. They get killed by it, so they have to use anaerobic respiration. When energy is in high demand, we have to use anaerobic respiration as well because there's not enough time to respire aerobically. And also when oxygen is unavailable, obviously we can't use aerobic respiration. So what happens is we go through glycolysis, the same as aerobic respiration that happens in the cytosol as well. Go through glycolysis, we get 4 ATP from that. So you might say, oh, what you said there were 2 ATP at the start. It's because we use up 2 ATP to get the reaction going. And then by the end, we're producing 4 ATP. So there's a net of 2 ATP. So we get 2 pyruvates from that. Those 2 pyruvates can go through two different pathways depending on what organism. So lactic acid fermentation, that happens in animals. Alcoholic fermentation happens in plants and yeasts. So lactic acid fermentation uses the NADH producing glycolysis to produce lactate, and the pyruvate is converted to lactate. In plants and yeast, the alcoholic fermentation produces CO2 and also produces ethanol. And each of these circles represents a carbon. Now the reason we use anaerobic respiration is because we want to get rid of these NADH and we want more NAD+, because NAD+ is required to break down glucose. So if we have too much NADH and not enough NAD+, we can't break down any more glucose. So we need to cycle it back to get more NAD+. All right, so factors that affect the rate of cellular respiration. O2 concentration, exactly the same as CO2 concentration. There's going to be limiting factors here that limit the rate of reaction. Glucose availability, exact same. And with temperature, it's going to be just like with photosynthesis, we reach a denaturation point. In humans, that denaturation point is going to be around 40 degrees. So our normal temperature is around 37, and then it starts getting a bit off around 39, 38, 39, around 40. That's going to be really dangerous for us. So endosymbiosis. Endo means inside, symbiosis means both benefiting. So endosymbiosis describes the theory that our ancestral eukaryotic cells were free-floating once upon a time, and they ate a bacteria. And that bacteria slowly became a mitochondria. So that's how, instead of digesting that bacteria, the mitochondria started working for our ancestral eukaryotic cell. And it started producing a lot of ATP for the cell. So obviously these cells are going to thrive because we're getting lots of ATP from the mitochondria, and we're also getting, and the mitochondria are getting protection, and they're getting food as well. And the same thing happened for chloroplasts. So the ancestral plant cell, after they diverged from the animal cell, they picked up some chloroplasts, and they can use that chloroplast to absorb light energy, convert it to glucose. And so they're both benefiting in that case. So we need to know about what evidence we have that this actually happened. This is a good mnemonic here that I found, Mad Doctor. We can look at the double membranes. So these organelles have two membranes, which is a lot like bacteria. And all the other organelles in our cells have a single membrane. So they're looking a little bit different. We can also say that the outer membrane belongs to the host, and the inner membrane belongs to the bacteria. All right, so antibiotics, they also are susceptible to antibiotics, which is the same as any other bacteria. They divide by binary fission, which happens in bacteria and not in eukaryotic cells. Eukaryotic cells divide via mitosis. They have circular DNA. Eukaryotes have linear chromosomes, but these bacteria will have circular chromosomes. Then they also have their own ribosomes, and the thing about those ribosomes is that they are a different shape and a different size to the rest of the cell. So that means they're producing their own ribosomes, and those ribosomes look a lot like bacterial ribosomes. So have a go at this question. All right, most people chose B, and that would be correct. This one was a little bit harder. So S is the double membrane, as we mentioned. That provides evidence for the bacterial origin of chloroplasts. V is your circular chromosome, and obviously that's the same in other bacteria. U would be, I think some people might have thought U was pointing to the chromosome. U is pointing to the stroma. So the stroma doesn't provide evidence of chloroplast bacterial origin. W are the ribosomes. That would provide evidence, but the thing is with U, it does not. T is just the outer membrane. I think that might have, or actually, I'd say S would be more likely to provide evidence. If you're just looking at T, if it only had T, then I wouldn't say it would provide evidence of bacterial origins. Cool. So have a go at this one. Have a think about your answer, and then I'll go through the answer. All right, so the thing is with these types of questions, there are two things that we need to address here. There's two different points that we need to address to get the 1 mark. We need to name a structural feature and we need to outline what it does or how it supports that theory. And the thing is with bio and with any other VCAA exam, you need to know what the verbs mean. So name means you just name it, you don't have to explain it. Outline means you do have to explain it. So you need to know those verbs. They have ribosomes, and that indicates they make their own proteins. We might have also said that those ribosomes look like other bacteria. And then you also have to bring it back saying they were likely once independent, or they were likely of bacterial origin, etc., etc. All right, we'll have a go at question 13. Is the poll working? Wait, can we see the answers? Okay, cool. I thought it broke. Never mind. So the answer was D. So below 10 AU, the light intensity is greater than the rate of photosynthesis. So if you can think about it as aerobic respiration is using up oxygen, so that's drawing oxygen into the cell. Photosynthesis is producing oxygen, so that's going to draw the oxygen out of the cell. So aerobic respiration brings the graph down, photosynthesis brings the graph up. Below 10, so this is the point of 10, that's when photosynthesis and aerobic respiration will be running at exactly the same rate. If the graph is coming down, that means aerobic respiration is going to be stronger than photosynthesis. At point T, photosynthesis is no longer occurring. It is occurring. It's occurring at the same rate as aerobic respiration. The optimal level of light intensity is 40. Not exactly. So it's still got a little bit to go once it's at that point. Point S, the amount of oxygen output is a third of that at point P. We can't exactly measure the exact amount of oxygen output because it's being counteracted by how much is being input in the aerobic respiration stage. So we'll have a go at question 14. All right, so the answer was B. Very good. So concentration of available carbon dioxide limits the rate of photosynthesis. So carbon dioxide is acting as our limiting factor from P to O. So P to O is where it plateaus and is the limiting factor there. Heat has denatured the enzymes. No, we're increasing light intensity, but light doesn't necessarily mean that our heat is increasing. Okay, even though light does provide heat sometimes, doesn't always. Light intensity has damaged the chlorophyll. If the chlorophyll had been damaged, the graph would have come down because photosynthesis would have stopped. So it wouldn't stay at the same point. High levels of oxygen produced at point P have accumulated, resulting in no more oxygen being produced. If no oxygen was being produced, then the graph would come down as well. So you just have to think about what are the different factors moving the graph up and down in each case. All right, another pop quiz. What is the best medical TV show? And bear in mind there is a correct answer. All right, so everyone is wrong. It is not Grey's Anatomy, it's actually Scrubs. Scrubs is the best medical TV show ever. And if you'd like to discuss that with me, you can stay after the lecture if you want. All right, so cellular signals. We have to know a bit about how different cells talk to each other. So the signaling molecules. Signaling molecules transduce a signal between cells via this thing called a ligand, and that is another word for signaling molecule. So they are received by a specific receptor. That receptor will only receive a certain type of molecule. And they move between cells to cause different responses to happen. The response from that cell can actually be dependent on the receptor and second messengers. So the same signaling molecule can produce lots of different responses depending on what receptor the cell has and what secondary messages the cell will have. So these are different types of cell signaling. Autocrine is when the cell targets itself. So it sends the signal and it's received by itself. Signaling across gap junctions is when it's kind of direct from the cytosol of one to the cytosol of another. Paracrine is when the cells are nearby and they're within the same tissues. And endocrine is when the signaling molecule moves into the bloodstream. That's a really important point. It goes into the bloodstream and acts on a distant target cell. So if it doesn't enter the bloodstream, it's not endocrine. So with animal hormones, they are produced by cells in endocrine glands. That is different to plant hormones because plant hormones don't have glands. Plants don't have glands. They don't have a bloodstream. So these are some of the differences between them. So instead of going through the blood, they travel through xylem and phloem. They're not produced in glands or specialized cells. They're not always a biomacromolecule. So a biomacromolecule is just a large organic molecule like a protein or a lipid. And they both have a target tissue that's distant to the source. So you do need to know a little bit about the plant hormones. You just need to know their basic function. So like auxin, cell elongation. Gibberellins got to do with seeds. Cytokinins, mitosis. Ethylene is fruit ripening. Abscisic acid is dormancy. So know a little bit about each and you can kind of guess from there. This hasn't really come up much at all in exams. Other signaling molecules that we need to know are neurotransmitters, and you also need to know about how what kind of signaling it is. So neurotransmitters transduce a signal between neurons. So they are also hydrophilic, which means they're received on the plasma membrane. Neurohormones are released into the bloodstream, and they act on a target cell. So neurohormones are released by neurons into the bloodstream to act on a target cell. And that means that they are endocrine. Cytokines are signaling molecules that allow communication between cells of the immune system. So cytokines, when you think of cytokines, think of the immune system. And they can be autocrine, paracrine, and endocrine. Pheromones are exocrine, which means they go out of the organism and act on another organism. And that's usually of the same species, which means they're conspecific. All right, so with neurotransmitters, we need to know about how they move across the synapse. So this is the synaptic cleft, and the synapse involves the two cells as well. That's the synapse. This is the synaptic cleft. So that's the space between them. So what happens is the action potential will come along. We don't need to know how the action potential works, but the action potential will come along to the axon terminal. And then once it reaches this terminal, these calcium channels will open and calcium will rush in. And what that calcium does is it stimulates these vesicles to move to the surface, and it'll expel its neurotransmitters to the receptors. So it'll diffuse across the synaptic cleft to the specific receptors. Then once they're received by these receptors, those receptors will trigger an action potential. And that leads to the continuation of the action potential in the postsynaptic cell. So this is electrical signaling through the action potential. This is chemical signaling across the synaptic cleft, and then we're back to electrical. With the stimulus response model, we don't have to know about homeostasis anymore, but we do need to know about these three steps. So reception is when the signaling molecule binds to the specific complementary receptor, and it causes it to change its shape. And that leads to transduction, signal transduction, which is where the change in shape of the receptor leads to a change in shape of enzymes within the cell. And that leads to a series of reactions which amplify and convert the signal to a form that the cell can respond to. And then it leads to a specific cellular response. All right, so with hydrophilic molecules, those are received on the plasma membrane, and that's because they can't pass the plasma membrane at all. Because remember, with hydrophilic molecules, they can't pass the lipid tails. Hydrophobic molecules, so steroids are one that you have to remember, are hydrophobic. They will pass into the cytosol, so they're received by a receptor in the cytosol, and they don't actually undergo signal transduction. So the middle stage here, that doesn't happen with hydrophobic molecules. Once it binds with the receptor, it might go straight to the nucleus, and it'll act as a transcription factor. So the receptor might be in the cytosol, the receptor might also be in the nucleus, so it can go straight to the nucleus. With hydrophilic molecules, we undergo all three of those stages. And this is just a little table with the main hormones in humans. So we've got amino acid derivatives and proteins, which are hydrophilic, and then we've got steroids, which are lipophilic or hydrophobic. All right, so apoptosis. This is natural, regulatory, programmed cell death. Those are the three key words that they have in the study design. So it's used when cells are supposed to die. And if we think about their purpose, it's usually if the cell is no longer needed. So like the outside layers of your skin, they're eventually shed off quite regularly. If a cell has lost its function, so if it no longer works anymore. The cell is a threat, so if it's infected or cancerous. If the cell is nearing the end of its life, you'll want it to die as well. And if the cell is damaged, you might cause apoptosis as well. So we need to know a bit about the steps involved. The actual phagocytosis of the macrophage at the end is not considered part of apoptosis. So don't include that in your answer if you're answering a question about the steps in apoptosis. So what happens is caspases are activated and they cleave proteins in the cell. Now, caspases are the basic things that cause apoptosis. Without caspases, apoptosis wouldn't happen. So caspases are little enzymes that go around breaking up proteins. That's what cleave means. So they cause the nucleus and organelles to condense, so they get smaller, and then they start fragmenting, which means they break up. The cell starts shrinking as well, and so it gets smaller, and then it starts blebbing, which means like little apoptotic bodies start coming off. So the vesicles start budding off, and the cell breaks up eventually. And then those apoptotic bodies are phagocytosed, which is different to necrosis, which we'll go through next, because in necrosis, nothing is cleared. So the phagocytosis of the apoptotic bodies means that there's no inflammation because all the debris is cleared by phagocytes. But remember, the phagocytosis is not a part of apoptosis. It happens at the end. So there are two main pathways that can trigger apoptosis. The intrinsic pathway is when the mitochondria senses something wrong within the cell. So it senses, okay, a virus has entered the cell, we need to self-destruct. And the mitochondria will release this thing called cytochrome C, which activates your caspases, and from there, there's no point of return. You can also get the extrinsic pathway, which is when another cell tells that cell, you need to die. So it sends it a signal via a death receptor, and you can also get activation of the mitochondria. So both pathways can lead to activation or breaking up of the mitochondria, and once the mitochondria breaks up, it releases cytochrome C, and that triggers caspases. You can also get direct triggering of caspases without the mitochondria. So we need to know about malfunctions in apoptosis as well. So that's when there's an imbalance in the rate of cell death. When apoptosis is at a very low rate, we can get tumors, which can eventually turn into cancers. So that's when cells are replicating way faster than they should be, and they're not dying. They're not responding to signals that are telling them to die. When there's too much apoptosis, we can get neurodegenerative diseases like Alzheimer's and Huntington's. So that's when cells in your brain or your neurons start dying when they shouldn't. So your brain will actually lose a lot of its cells. Necrosis is uncontrolled and unplanned cell death. So we don't want necrosis to happen, and we can't control what happens in necrosis. And it leads to inflammation, which is very bad, because all your cell contents aren't being contained in those little apoptotic bodies, and so it's causing damage to all your other tissues. So in necrosis, instead of shrinking, your cell will actually swell and burst, and that spills all of the contents all over your tissues. And that leads to inflammation. So in terms of the differences with apoptosis, it leads to inflammation, it's unwanted, and there's no apoptotic bodies that are cleared by phagocytes. Have a go at this question now. All right, so the answer was D, which most people picked. I think B was another popular answer, but in terms of decreased rate of apoptosis, that is actually only happening to the cancer cells. The surrounding cells are perfectly fine. They don't have any decreased rate of apoptosis. All right, so responding to antigens, now we're getting a little bit into immunology. An antigen is an identification molecule that can initiate an immune response, and in particular, production of antibodies. So anti means antibody, gen means generator. So antigens will lead to the production of antibodies. A self-antigen is made by your own body. Non-self antigens are made by something else. And an allergen is a substance that produces a vigorous and hypersensitive immune response. So something that triggers a response which you don't want, and which will lead to a response that actually causes harm. Pathogens are disease-causing agents. So in terms of your non-cellulars, you've got virus and prions are just malformed proteins. Unicellulars, bacteria, fungi, and protists, and your multicellulars are tapeworms. So prions, as I mentioned, they're malformed proteins. They can malform other proteins, and they're resistant to enzymes and heat in your body. Your viruses are non-living, and they're non-living because they can't replicate by themselves. And they have a protein capsid around them, they have genetic material, they have antigens, and they might have a lipid membrane. And you do need to know about their structure as well. So they're not considered living because they're not made of cells, they can't reproduce by themselves, and they can't undergo intracellular processes. Bacteria are intracellular or extracellular prokaryotes, so they can reside inside your host cell or they might not. Fungi are eukaryotes, and in terms of the innate and acquired immunity, so these are your two main sections of immunity. Your innate immunity is will lead to the same response to every pathogen every time. So there's no memory cells produced. With your acquired immunity, you do get memory cells produced, which means your response will be a lot stronger the second time around. The first line of defense prevents entry of organisms into the host. So it's all these things here, which I have in my notes as well. The second line, and it's a bit different for animals and plants. So plants have, different barriers. So they've got physical, physiological, chemical, and biological for both, but the barriers are a little bit different. And I think we'll skip over this just so we can answer some questions at the end. In the second line of defense, this is the innate response to infection. So we're not producing any memory cells in the second line of defense. And that involves all of these cells here, which are macrophages. Those are a type of phagocyte, and they are antigen-presenting cells. So they phagocytose a bacteria or a virus, and they put their antigen on its membrane, then they take it to T cells, and they tell the T cell, okay, this is what I found, we need to produce a response against this. That's what an antigen-presenting cell does. And your dendritic cells are the other type of antigen-presenting cell. In terms of VCAA, I think they want you to know mostly about dendritic cells. So if we mention APCs, most likely we're going to mention dendritic cells, but they can also be macrophages. Neutrophils are the first cells to reach the site of inflammation. So they're granulocytes, which means they release chemicals from your granules, which can break down other pathogens. They're also phagocytes, so they engulf or eat other cells and viruses. Your eosinophil is a granulocyte, and they're mostly used against multicellulars, so things like worms. Your natural killer cell is a granulocyte as well, so it signals other cells to die. And that only happens when your target cell, your self cell, is not displaying the right self antigens. So natural killer cells only kill self cells that are not displaying self antigens. Mast cells release histamine, and that increases inflammation, and platelets are involved in blood clots. In terms of the humoral second line of defense, so humoral means it's got to do with the humors of the body, so the fluids of the body, all the molecules that float around in the fluids. The cellular second line of defense has got to do with all the cells involved. So histamine does all of these things, which is in my notes as well. So it increases inflammation through dilation of the blood vessels, increased permeability of the blood vessels, and that increased permeability allows the cells to squeeze through, so phagocytes. Your complement proteins do a lot as well. So they are kind of like innate antibodies. So they bind to the outside of your pathogen, and it'll signal other cells to come and attack. It'll lyse the membrane of your pathogen by forming a membrane attack complex. So it can actually poke holes in the membrane, and then it can clump all these bacteria together as well. Your interferon is a type of cytokine that acts against viruses. So it signals your cell to be virus resistant. So interferons are against viruses. That's an important thing to remember. They can prevent assembly of viruses within your host cell. They can trigger cells to undergo apoptosis if they have been infected, etc., etc. Your cytokines I've mentioned can do lots of things. They do lots of different signaling between cells of the immune system. So they can increase fever. They can tell cells where to go. So increase chemotaxis, which is when you tell cells where to go, pretty much. And in terms of the inflammatory response, this is the increase in the response of the second line of defense. So initially we'll have a wound here, and your bacteria will come in. The mast cells, which reside in your tissues, will send out histamine, and that leads to dilation of the blood vessels, increased permeability. And that increased permeability means your neutrophils can come in and start phagocytosing the bacteria, so start killing the bacteria. Your macrophages will also come along. They'll engulf some bacteria, and they'll start displaying the antigens on their MHC. And they'll also release cytokines, which will signal to the rest of the body that we're finding a pathogen. With the lymphatic system, this system is made of lymph vessels and lymph nodes and lymph organs, and they pretty much drain all your fluid, your excess fluid, from tissues in the body. They also filter that fluid, so they get rid of pathogens, and they also hold your lymphocytes. So your lymphocytes are your T cells, your B cells, and your natural killer cells, and they hold them in your lymph nodes. Primary lymph organs are where your lymphocytes will develop and mature. So that's the bone marrow and the thymus gland. Your secondary lymph organs are your lymph nodes, tonsils, and your spleen, etc. So the third line of defense consisting of B and T cells. So this is where we get specific. So this is acquired immunity. They like to chill in the lymph nodes until they are activated. So when they're presented with an antigen by an antigen-presenting cell, they will become activated, and that's when they start doing their functions. So plasma cells, B cells will differentiate into plasma cells and memory cells. So the plasma cells produce lots of antibodies. Your T cells, they can either be T helper cells or cytotoxic T cells. Your T helper cells control the rest of the immune system. Your cytotoxic T cells will kill self cells that are displaying bad antigens. So your natural killers kill self cells, and your cytotoxic T cells kill self cells as well. And the difference is that cytotoxic T cells are specific, your natural killer cells are non-specific. All right, so in terms of activating your B cells, first they develop and mature in the bone marrow. And that's different to T cells because T cells develop in the bone marrow and they mature in the thymus. So when a naive B cell binds to a specific antigen, it'll differentiate into plasma cells and specific memory cells. And that only happens if it's activated. Sorry. It only happens if it's activated by T cells. And it'll produce plasma cells, which produce antibodies, and memory cells, which keep a memory of the antigen for next time. And the memory cells can later differentiate into plasma and memory cells the second time around. Your T helper cells are the big brain of the immune system. They control other components, and they stimulate B cells to proliferate. They stimulate cytotoxic T cells, and they are specific to one type of antigen. Your cytotoxic T cells, as I mentioned, they kill specific self cells that are displaying bad antigens. Through antigen presentation, this is how we get the antigen to your lymph nodes. So your macrophage or dendritic cell will engulf the bacteria or virus. It'll form this phagosome, so that's just the vesicle in which it holds it. That phagosome will combine with the lysosome to form a phagolysosome, and that breaks up the bacteria, and it produces these antigens, which it displays on MHC2 on its membrane. All right, so in terms of using the antigen presentation to activate the third line, we get these dendritic cells picking up, or macrophage picking up those antigens. It brings it to the lymph node where a specific T helper cell will sense that antigen. And then we also get, at the same time, a naive B cell. It comes along, it detects an antigen, and it also displays an antigen on its membrane. So a B cell is also an antigen presenting cell. And the thing is, with those B and T cells, they are specific to one type of antigen. They don't work with any other antigen. And there's millions and millions of B and T cells residing in your lymph nodes. You need to find the right one. So the macrophage has to go around and find the right B and T cell. And once the T cell is activated and the B cell is activated, the T helper cell will activate the B cell again, and it'll proliferate and produce more antibodies.
Ethan, I think you've got your VC assessor on.
Oh, no worries.
Diane, cool. Is that you? I think you're on mute still.
There, how's that?
Hi, Diane.
Hi. I've never used this platform before, so
that's all right.
Can you hear me okay?
Yeah. Oh. Is it all right if I just go through these last couple of slides? I've got 4 more to go.
Heaven forbid I should stop you in the middle of specific immunity.
No worries.
Go for it, yeah.
Cool, thank you. Okay, so We're actually going really good for time, which I'm really happy about. Okay, so in terms of activating your cell-mediated third line of defense, I couldn't find a good diagram, so I made a little crappy diagram here. Basically, your virus-infected cell will display an antigen to a cytotoxic T cell, and that tells the cytotoxic T cell that something's wrong with it, but it hasn't been activated yet. Then your dendritic cell will come along, display an antigen to a specific T helper cell. And that T helper cell will say, okay, I need to activate some cytotoxic T cells because we're seeing that there's this virus that's infecting our body. So if it's a virus, we're more likely to use cytotoxic T cells. If it is a bacteria, we're more likely to use B cells. Then the T helper cell will use cytokines to stimulate the cytotoxic T cell, and then that cytotoxic T cell will release perforin and gramzymes, which induces apoptosis in the infected cell. All right, so in terms of your antibodies, they have a constant region, which is this part of it. And that region stays the same, doesn't matter what antibody you're using, unless we're talking about different isotypes, which we don't really need to know too much about. And your variable region is this N part, and that's the part that binds to your different antigens. That will be complementary to each of your antigens. And that depends, the same antibody is produced by the same plasma cell. So the plasma cell is specific for one type of antigen, and it'll produce antibodies that bind to that one type of antigen. So they can neutralize the pathogen, which means they just bind to the outside of the pathogen and prevent it from causing harm. They can opsonize, which means that it allows phagocytes to eat it more easily. They can agglutinate pathogens, which means they clump them all together. And they can activate complement proteins, which leads to the membrane attack complex. And this is just a quick slide about how the primary response is not as strong as a secondary response, and that's because we have memory cells in the secondary response. So it'll be faster and stronger. And those are the keywords you need to have in your answers. All right, so might skip those two questions. We have a VC examiner with us today, and I might go to Mentimeter, have a look at the questions in that. So we'll open up a Q&A for Diane to help us with. So if anyone wants to go to this site, ask a few questions, I'm sure Diane would be happy to help us out.
How do I know the questions?
All right, so, oh, sorry. I'll read them out for you. So, we'll start with the first one. Okay, hang on.
Ethan, it might be easier if you share the screen for Mentimeter so people can see it as well.
Sure, cool. So, Diane, do you have any strategies to tackle the exams?
Okay. Now on my screen, I've got sorry for my lack of ability here with
That's
Zoom.
okay.
Zoom before. So on my screen, there's a bit on the right that's got a picture of you and a picture of me and someone called Elise. Hi, Elise. And it's covering up part of the questions. So
okay.
Move that out of the way. Can I just... Oh,
you should be able to drag
I've figured it out.
Cool, no worries.
Okay. I'm a bit of a dinosaur sometimes as well. So.
That's fine.
Okay, so what strategy do I have to tackle the exam, I assume that means?
Yes, I think it's exam, yeah.
Okay, and you know, I was asked this very same question by one of my students the other day, and it always fills me with fear when I hear a student suggesting that they leave the multiple choice till last. I would never, ever do that.
I agree with that.
My strategy that I always recommend to my students, and I just thought, I don't know, have you guys told the students anything about me, or would you like me to do a 30?
Oh, you go for it. Go for it.
Yeah. So I've been teaching senior biology almost exclusively since 1982, and I've been an assessor of the exam on and off throughout that period. So just to give you a bit of background on the fact that I've got a little bit of an idea of what I'm on about. And also, can I say what school I'm at, or?
Sure, I think
Yeah,
so.
yeah. So I'm a senior biology teacher at Nossal High School. I've been there for this is my 10th year there, and yeah, basically almost exclusively teaching VCAA biology for that entire time. So that's probably all you need to know about me. So when it comes to doing the exam, what I always tell the students is to use their reading time as effectively as possible. So as soon as the invigilator says you can start reading now, you should flick straight to section B, which is the short answer section, and start getting your head around what each of the short answer questions are actually about. So there's usually 11 short answer questions of varying lengths. They can be as short as 3 or 4 marks and as long as up to maybe 12 or more marks. So they really do vary in size. But I strongly urge you to try and get your head around, for example, what topic is each of those questions on, and I can talk a little bit more about that shortly. And that just basically starts to help you feel comfortable with the exam and familiarising you with what's there. And if you're well prepared, there shouldn't be anything too scary there. Now, VCAA do state every year that there may not necessarily be the same number of questions every year, but for the last 3 years, there's been 11 short answer questions. That's section B, of course. Section A always has 40 multiple choice. So getting back to your strategy, so you read the short answer questions as thoroughly as you can in the 15-minute reading time that you get, so that you've really got an idea of what's coming. The other thing about that is that I always tell my students to read the whole question. When it's a short answer question, read the whole question through at least once before you start answering, because often students will overdo it in part A and then get to part B and think, oh, I've already answered that. So you need to have a look and see what's coming so you can plan how you're going to approach that question and what you think needs to go into each sub part of each question. Then any time left over in your 15 minutes reading time, go back to the multiple choice and start reading those. When it's writing time, do the multiple choice first, because that's when you're really fresh. And, it's a 2.5-hour exam. It's a long haul. So multiple choice, there's a lot of information in there. There's A, B, C, D for every question. There's background material you've got to analyse and synthesise, and there might be tables and graphs and all sorts of things that you've got to work your way through. So you really, really need your brain to be totally switched on at that time. And you should be able to get through those 40 multiple choice within about 30 minutes. That's what I find with my students. That will then give you 2 hours to do the short answers, and that's super important. I'll come to that in a moment. Now, with the multiple choice as well, as you're working your way through the multiple choice, I can tell you for a fact that the examiners are not going to have any answers to short answer questions embedded in the multiple choice. They make sure they don't do that. They're not kind in that way. But there are many, many things within those multiple choice that can act as what I would call memory triggers. And so little terms that will sort of like open up a file in your brain and you'll go, oh yeah, all that stuff I learned about complement proteins or whatever. So the multiple choice is really a good way to stimulate your cavernous brains and get you really opening up all that knowledge that you have and getting ready for the short answers. Now, statewide, every year, VCAA do a little bit of a statistical summary of how students have performed. And we usually have around about 15,000 students sitting the biology paper every year, roundabout. And what they tend to find is that the average mark, that's not what you guys are aiming for, I wouldn't think, but the average mark for the multiple choice is usually around about the mid-60s%. So around about 65% would be the average score for the multiple choice section. However, the average score for the short answers are usually just under 50%. Now that's a huge difference. And for ambitious students, which I'm assuming you guys are, you would sit up and pay attention to that piece of information, because that would obviously make you think, well, if I do really well on the short answers, then I'm going to be, you know, pushing myself further and further up the rank order, if you like. So you need to allow yourself as much time as you can, not to the detriment of your answers in section A, of course, but as much time as you can for the short answers. And then with the short answers section, timing is also extremely important. So you should be watching out for how much time you spend on each question, because there's nothing worse than getting to the end of the exam or the end of the time and running out of time, and you've missed out on answering a question that you could easily have done, but you spent too much time on the other questions and you've run out of time. Do the questions in the order in which they appear is always my advice to students, because if you think, oh, question 3, that's an easy one, I'll leave that and I'll go on to question 6 because that's a really hard one, and I'll come back to question 3. And the number of students that forget is also really, you know, a bit heartbreaking when students walk out of an exam and either say, I ran out of time, or I forgot to do question 3. So in a nutshell, they would be my main strategies for exam completion.
Hey, Diane. That was really great. Ethan's having a little bit of a technology issue, so he's going to be 5 minutes, but I'm just going to take, I'm going to ask you a couple of questions, some questions that students have asked. I've got one right here. It says, how do you suggest we go about the biology exams? Should I just do practice exams from the last 10 years, or should I go through content from the textbook as well? What do you suggest?
Okay. So I'm assuming that student means preparation for the exam. Yeah. Well, I mean, the current syllabus came in for Unit 3-4 in 2017. So doing past exams for the last 10 years is not necessarily that helpful. You have to be careful. Anything that has the date 2016 or earlier on it, you have to be very mindful of the fact that the syllabus changed quite a bit. And for example, all of the good old-fashioned traditional Mendelian genetics like Punnett squares and pedigrees and all of that used to all be in 3-4, but they've taken that out since 2016. They've taken all of that out and put that back into Unit 2. So when you're doing those practice exams or those past year exams, just be mindful of that if you're doing anything from before 2017, there are going to be questions that test content that's no longer in the Year 12 exam. And there'll also be, there's been a change in style as well, in the way in which they ask questions. So I could elaborate on that more if you like, but before I do, I could also suggest that students try to get their hands on trial exams from the various different companies that produce those exams. So am I allowed to name companies, or is that?
Yeah, of course.
Yeah. So every year at my school, we always buy, because these things are copyright, so we always buy exams from NEAP, NEAP, from TSSM, from Insight. They would be, for me, the top 3 that I would recommend students do. And then if they can get their hands on them, anything from STAV, the Science Teachers Association. And then there's also another company that calls themselves Learning Materials. They're quite good. They're a bit more sort of basic, but if you're a bit shaky in an area, you might want to look at questions from those ones. So, and also 2 of those companies, TSSM and Learning Materials, produce topic tests. So, you know, a student might just have one area of weakness. Perhaps they were sick, or they were away, or they never really quite got their head around the immune system, for example. So they could just, you know, spend a bit more time practicing those. As for going back to the textbook, I always say to my kids at this time of the year, don't do that. Because at this stage of the year, the only way you're going to know if you're ready is to actually do practice exams. And from those practice exams, you should then be able to identify if you have any areas of weakness regarding content knowledge. And that's when you go back into your textbook, or back into your notes, or back into the PowerPoints that your teachers have given you, and look at those. So, for example, the last couple of days I've had questions from students, and my answer, interestingly, has been to all 3 of them. why don't you take a look at the PowerPoint that we had for that topic? I think you'll find the answer in there. So, you know, your teachers prepare all sorts of resources, all sorts of wonderful resources, and you'll often find what you're looking for is in there if you think you've discovered an area of weakness. So I could say
yeah,
a lot.
that's
Yeah.
because I teach mathematics, and we often don't recommend other companies because they sometimes stuff things up a little bit. But for biology, it seems like you would really recommend.
Yeah, look, you're right. The companies do sometimes stuff things up, and I find that NEEP is the most reliable, although it will sometimes push the students a bit beyond what's actually in the syllabus.
Yeah,
But
definitely.
what I also find is that when students come across a question where you know, the person writing the exams, I'm sure has been very well intentioned, but has written a question that's not really on the syllabus, or a question that's ambiguous or lacking in enough background information to even be able to answer it properly, then that's where your teacher comes in. And, you know, you just screenshot it, send it through to your teacher and say, what's going on with this question? And then you have a really good discussion about it, and the students actually learn stuff from that. They become even better at doing what they're doing because they've had to go through that whole analysis. Why was this question wrong type of thing? So yeah.
Yeah, definitely use your teachers. I would definitely recommend that too.
Yeah.
Looks like Ethan, I think you're back now.
I'm back. My Zoom crashed.
Yeah,
Sorry, everyone.
That's okay. We just went through one or two questions about how to study for the exam and stuff like that. But yeah, you take it away. Good luck.
Cool. I'm not sure if this has already been answered, but Diane, what do you think about this question? How do I gain full marks in short answer?
Well, I wish I could sit next to you and whisper the answer in your ear. I have a thing that I call question attack, and question attack has three aspects to it. One is how many marks is it worth? Two is how much space have they provided for your answer? And three, and most importantly, is what are the command terms? So I'm going to assume that your students know what I mean by command terms, or would you like me to explain that a little bit?
Yeah, the only thing I've said is like know the verbs in the question, but could you please explain it like a bit better?
Okay. Well, at our school, we call them command terms. And so we're talking about things like identify, justify, list, describe, explain, those sorts of terms. So we call them command terms because they're basically commanding you what to do in your answer. And, you know, one of the biggest issues I see with students when they're preparing and when they're doing SACs, for example, is that they just don't get the difference between a describe question and an explain question. So I spend quite a lot of time with my students looking at examples of that. And for example, if you get a question that says what or how, that's a describe question as well. So they might say what in the question, or they might say how does that work, for example, or they might ask you to describe. But in any of those three ways, they're pretty much asking the same thing. And describe means tell me what you see. So often at 3-4 biology, we're working at the molecular level. So if you were asked to describe how a cell identifies a particular ligand, for example, a particular signaling molecule, then you would have to force your brain to go to the molecular level and describe what that would look like. So in other words, the complementary shape of the receptor molecule and how the messenger molecule slots into that receptor molecule. So that's a describe, that's a what. But then if it asks you to explain, then that wants deeper thought. That requires deeper thought. That requires you to bring in some extra biology theory into your answer, because explain means why. So sometimes in a question they'll say why, sometimes they'll say explain. Those two things basically mean the same thing. So watch out for the command terms. Practice different types of questions that use different types of command terms. Pay attention to how many marks the question is worth. If it's worth 3 marks, for example, then make sure you've got 3 solid points that are different from each other. Students often write 2 things, think they've written 2 different things, but they've actually written the same thing twice, but just in a different way. So just try to imagine how the person marking your exam, the assessor, how they would be deciding whether or not to give you those 3 marks. And they will be definitely looking for 3 distinctly different things. And pay attention to how much space they've given you as well, because if it's a 1-mark question but they've given you 4 lines, then obviously they want a little bit more detail than just a single word or something like that.
Definitely.
I mean, there's a lot more I could say, but I think that's a pretty good summary for that question.
Cool. Thank you. I'll just look for another question. I think we've kind of covered timing on the exam. So I think you said a little bit about going multiple choice first is always a good idea.
Oh, definitely.
Can we write it in dot points for short
Oh,
answer?
you know, I was just thinking if they don't ask me that, I'm going to say it anyway. So imagine your assessor. Your assessor could be some old duck like me who's foolishly put her hand up, you know, again, another year to mark the exams. And trust me, you don't do it for the money because the money is horrible. You do it for the experience so that you can bring that experience back into your own classroom for your own students. And so I've been given a very short space of time to mark 300 exams. Okay. So you can imagine that after the first 50, you get on a bit of a roll, but you're also thinking, oh God, you know, am I ever going to get through this? So be kind to your assessor. All right. Don't write a whole lot of waffle. Please, please, please write your answers in dot points. I tell my students dot points all the way, and they don't believe me. It takes me months to convince them that we don't need complete, beautifully constructed, grammatically correct sentences. We're not looking for that on the biology exam, not by any means. And don't let anybody else tell you otherwise, okay? We want dot points, we want concise, we want scientific language, we want scientific terms used in the appropriate way. Use the best science words you can. And definitely dot points all the way. Yeah.
100%. Cool. Yeah, I think this isn't an arts subject, so we don't have to make lovely paragraphs for our answers.
No topic sentences. Definitely not required.
Cool. Let's see. How strict is the marking rubric for assessors? On short answers, is there a list of acceptable answers, or is it up to the assessor's initiative?
Okay, that's going to depend wholly on the question. So sometimes with questions, they might be asking you, for example, for an ethical implication of a particular scientific technique. So a student might come up with something really different that the examiners didn't think of when they were making up the marking scheme. And yet it could be a really, really good answer. So the assessor does have the discretionary powers to accept a slightly different answer in some circumstances. But for the most part, the assessors are instructed to stick to the marking scheme really, really strictly. And the reason for that is because they want all the exams to be marked to the same standard. Otherwise, it's not fair. And in fact, when you're marking, you do your marking online, and every now and then they pop in a paper. You don't know it's happening, but they, like me, I don't know it's happening, but they've popped in a paper that's actually already been marked by the chief assessor. And if my mark is different from his mark, then I will be flagged and I will be contacted, and they will say, what are you doing? You're not doing it right. So they're actually really quite strict. And every exam is marked twice. And the computer, so by 2 different assessors, and then the computer compares the mark from one assessor with the other assessor. And if it's more than a few marks difference, then it's sent to a 3rd assessor. So it's actually, I've always really admired the way this process is done, because it's very, very fair. It's very democratic and it's very fair. But it does mean that we have to have a really good, as your student asked, rubric. It's not a rubric per se, it's a set of instructions, but we go in for a whole day, what they call assessor training day. We go into Science Teachers Association headquarters, and they train us up for a whole day on how to mark this exam, and we nut out any issues that we think we're going to have, and we ask questions, and we just make sure that we're really clear in our minds how to mark it so that everyone will be marking it the same way.
Cool. Thank you so much, Diane. One more question,
Sure.
And then I think we'll get back to Unit 4. This person says, I didn't do that well in SACS. Have I screwed myself over? I get this question a lot from my students, but what do you think, Diane?
I just did 56 parent-teacher meetings last week, and I got that
So
question.
they all ask the same question, yeah.
I got that question a lot. And one of the things I said to my students was, every year VCAA will standardise your SAC performance against your exam performance. Okay, so they'll look at how you and your the other students in your cohort performed on the SACs, and then they'll look at the same thing for the exam. And if the SAC scores are a lot lower than the exam scores, then they will bring up the SAC scores. Now, that happens in a normal year. This is not a normal year. So I believe, this is my theory, I believe that this year more than ever before, they're going to have to do that, because they know that the exam is a reliable assessment tool. And if you're a well-prepared biology student, you should know what we mean by reliability and validity, right? So they know that the biology exam is a reliable and valid assessment tool. But as far as SACs go across all the different schools, they've got no way of knowing that. So the short answer is no, you haven't screwed yourself over, because the exam is 60% of your study score, the SACs are 40%. And if you absolutely smash that exam, you will pull yourself right back up that rank order.
Absolutely.
So go for it, guys. Be really well prepared. Yeah.
You can always pull yourself back
Yes.
in your exam. Always, always. One more question, because this one got a lot of likes. Is it really hard to get above a 35 study score?
Not to get above 35. It's very, very hard to get above 45. And it's quite tricky to get above 40. So yes and no. I mean, 35 is your midway, really. So, well, maybe I've got a bit of a skewed idea because of where I teach. You know, kids get below 35, they think that's the end of the world at my school. But
it really depends on on your own circumstances.
It's not super hard, and if you're really well prepared, you should be able to get, you know, high 30s or low 40s. And of course, if you're one of those top kids, you could get a high 40s. But, you know, that's a really, really difficult question to answer, because I'd need to know you a bit more personally to be able to answer that question. Yeah. I'd need to know a lot more about your own personal circumstances to be able to answer that. I'm sorry.
Cool, no worries. So yeah, I think I don't know, in my experience, I feel like a lot of it's got to do with how much you prepare, but there is also a little bit of chance on the day.
Yeah, there is chance, because they always throw in those curveball questions where you go, whoa, where did that come from?
Yeah.
So you just got to be really, practice some mindfulness before you go in. Don't do that whole thing with your students. Oh no, I'm so scared, with your friends rather. I'm so scared, that sort of thing. Just be calm and have a good night's sleep the night before, and have a proper breakfast, and no Red Bull, and, you know, all the things that your mother tells you not to do. And it
Definitely.
It does come down to how you perform on the day. So there can be chance involved.
Yeah, and so I feel like that's really important to remember, so you don't put too much pressure on yourself.
Yeah.
Because if you realize that there's some things that you can't control about what's going to happen on the exam, you're not going to stress about it.
Yeah, and just do the questions that you know you can do, and do them really, really well. There's nothing an assessor likes more than seeing a competent answer where the assessor can go, oh yeah, this kid really knows what they're talking about, 2 out of 2, 3 out of 3, as opposed to students who are just waffling all around the point, and the assessor's looking at it and going, really, what are you going on about? So yeah. Do the ones you can, and do them well. Yeah.
No worries. Okay, so we might finish the Q&A there. Thank you so much to Diane. I learned a lot from that as well. I will pass that on to my students as well. Thanks, Diane. We might get back to Unit 4 if that's all right.
Yeah, of course. My pleasure.
No worries.
Good luck, guys. Good luck on your exam.
Thanks.
Oh, you're welcome, Janice. Bye.
Cool. See ya. All right, so now we'll just have a short video from UBS, our sponsor, and it's just a little bit about how some of their volunteers found high school. So just plop this one up. It's just loading. All right.
Hi, I'm Boswell and I'm currently a third-year cadet at UBS. My role is a quantitative analyst in the equities division and my responsibilities involve algorithmic trading and data analytics. My biggest career highlight has been the opportunity to learn a lot about both the business and technology as it ties in directly with what I'm currently learning at university. I study maths and computer science, and during high school I really enjoyed maths and physics. The advice for students sitting the exams is to just relax and don't let your results define you. If you've already put in effort throughout the year and consistently studied, then you're pretty much guaranteed to do well.
No worries. Cool. All right. So thanks to Boswell for making that video. We might continue on now. We've got a quick pop quiz here. So Andrew, if you could pop up the multiple choice. What's this guy's name? Right, if we could have a look at the answers. The results. Cool, so it was Yondu. Good. Django is from a completely different movie. This guy is from Guardians of the Galaxy, which I recently watched. All right, cool. Immunity. Types of immunity, we've got, we can split it into active, passive, and natural and artificial. Active is when we're making memory cells. Passive is when we don't have memory cells, so when we've just got antibodies. Alright, so natural active is when we're encountering the virus ourselves. So natural means it's not got to do with human-made stuff, so encountering the virus or pathogen ourselves, so it's natural infection. Natural passive immunity is through breast milk, so we're receiving antibodies from our mother, or through the placenta before birth. Artificial active are things like vaccinations, so that's when we give a person an antigen. Passive artificial is things like anti-venom. So we actually give horses a venom, and since they're so big, they're not affected by the venom that much. So they produce lots of antibodies, and we can take those antibodies from the horse and then stick it in people who have been bitten by a snake. And so that's just passive immunity because we're not producing memory cells. But with vaccinations, we are producing memory cells. So that's why we call it active. So vaccination exposes the immune system to antigens found on the pathogen so it can produce memory cells. So we do this because we need to be able to produce a stronger response the second time around, and it needs to be much faster, and that's what gives us immunity. Why do we need boosters? And that's because for some vaccinations, the memory cells actually die over time. So that's why we need boosters, and that increases the number of memory cells so that it becomes even stronger second time around. Herd immunity is the concept where if a large proportion of the population is vaccinated, there are fewer people to get the vaccination from. So the people who are vaccinated kind of provide a barrier. So you can see with this diagram here, with no immunization, this virus can spread to this person, then to this person, then to this person, and can reach everyone pretty much. With vaccination, the susceptible people, so this person is directly next to a contagious person, so they can get the virus, but you can see that all these other susceptible people are kind of protected by the immunized people. So usually we aim for around 95% immunization. And obviously there's cases where people can't be immunized if they're, for example, immunocompromised, or if they are exceptionally old people or exceptionally young babies won't be able to be immunized as well. So parts of the immune system that can malfunction, we need to know about autoimmune disease. And that's when the immune system starts attacking your own body. So it identifies self-antigens as non-self, and it starts attacking it. Now, VCAA wants us to know about multiple sclerosis. And that's when your immune system sees the myelin sheath around your nerve axons as non-self, then it starts attacking it, usually with cytotoxic T cells, but a little bit with B cells as well, which will produce antibodies. And that means that slowly this person will lose nerve function because the myelin sheath insulates the axon and it allows nerve impulses to pass through. Without a myelin sheath, your neurons will conduct signals much more slowly. So this person will slowly become paralyzed. We've discussed herd immunity. Oh, go backwards. Okay, so with immune deficiency, autoimmune disease was a hyperactive immune system. Immune deficiency is an immune system that's not working as well as it should. So that happens in AIDS, acquired immunodeficiency syndrome. HIV is the virus that causes AIDS. AIDS is not a virus, AIDS is the result of HIV. HIV targets immune cells, specifically T helper cells. And as we mentioned, T helper cells control all the other cells of the immune system. So if your HIV is killing T helper cells, you can imagine that your immune system is not going to be doing too well. And so the immune system can't fight infections, and it'll start succumbing to very common things that we shouldn't be getting disease from. So if people with AIDS, it's very sad, they can be killed by the common cold because they don't have T helper cells. With allergic reactions, this is obviously with a vigorous and hypersensitive immune response to something that shouldn't be responded to, so things like pollen. With your first exposure, that leads to the production of specific IgE antibodies. That's just a type of antibody. And those IgE antibodies will bind to the membrane of mast cells. And now these mast cells are called primed mast cells. And the second time we encounter pollen, second time we encounter an allergen, the allergen will come and bind to both of these, to two different antibodies, and that's called cross-linking. And then that triggers histamine to be released from the mast cell, and that triggers the immune response. So things like bronchoconstriction, rash, heat, hives, that kind of thing. Monoclonal antibodies are lots of one specific antibody. So mono means single, clonal means replicating. Lots of one specific antibody, and they're all artificial, so they're made in a lab. Okay, we won't go through how they're made. So, Unit 4. We need to know about the types of mutations. We've got point mutations, so these ones on the left, and then we've got block mutations, the ones on the right. In your point mutations, you've got silent mutations, so that's when the amino acid stays the same. And that's because of the redundant nature of the genetic code. So the same, different codon codes for the same amino acid. That's a silent mutation. With nonsense mutations, that's when this change of base has led to the formation of a stop codon. So instead of coding for a codon, it tells the ribosome, you need to stop here, and that means our peptide is only 2 amino acids long. A missense mutation is when the base changes and you get a different amino acid, and that can be either conservative or non-conservative. A conservative missense mutation is when the function of the protein stays the same. Non-conservative is when you lose the function of the protein. So even though the amino acid has changed, you can still maintain its function if the amino acid is similar enough to the original. A frameshift mutation is when, because the DNA is read in groups of 3, or the RNA is read in groups of 3, if you lose one base or you add a base, those groups of 3 will all be shifted. So now we're doing the first part of this codon and the 2 bases of this codon. So we're actually getting completely different codons everywhere after that mutation. With your block mutations, you can get deletion. So a block mutation first is a change to 2 or more bases in a DNA molecule. Deletion is when you delete an entire section. Duplication obviously is when that section is duplicated. Inversion is when you take out the section, you flip it around, and then you put it back in. And that can be devastating. If there's a gene that goes across that section, that gene is going to be cut in half. So now half the gene is here, and half the gene is here. So that gene has been destroyed. And insertion is when you move a section from one chromosome to another. Translocation is kind of similar to insertion, except instead of moving, they swap. So those 2 sections swap. We need to know about different things that can change allele frequencies as well. So selection decreases genetic variation. Gene flow can increase genetic variation, and genetic drift decreases variation. So the difference between a selective pressure and a selective advantage is that a selective pressure is a feature of the environment that favors certain individuals over others. A selective advantage is a feature of an individual which makes it better for survival compared to others. So these are the steps that you need to know in natural selection. You need to state that there is variation in the population, and you need to specify what kind of variation it is. So if we're talking about these giraffes, there's variation in the length of their necks. Some individuals have a selective advantage, so these ones can reach the leaves. In response to selection pressures, that would be high leaves. Therefore, they're more likely to survive and reproduce, so over time the allele frequencies increase and the population evolves over time. Gene flow is the migration of alleles between populations. So we can see with this population, it doesn't have any brown phenotypes. And now since this beetle has moved over to this population, the genetic diversity of this population has increased. So it's going to start interbreeding with these, and then now we're going to get a new allele in this gene pool. Genetic drift are chance events that result in changes in allele frequencies. And that makes it different to natural selection, because with genetic drift, it's all up to chance whether you survive or not. In natural selection, it is dependent on your phenotype. With genetic drift, it's completely random. So in founder effect, that's when a small section of the population is taken to a new area, or they migrate to a new area and they start a new population. So you can see that these allele frequencies are actually very different to what they started off with, because there's no reds that have moved over to the new population. With a genetic bottleneck, that's when they stay in the same spot and a large proportion has been eliminated, which means that the new population will not be representative of the original. And what I mean by representative is that we've got 50% black, 50% red, and now we've got only 2 blacks and 10 reds. Alright, so allopatric speciation is another thing that pops up a lot. And you do need to know these steps. These are the main 3 steps that you have to have, and then step 7 as well is really important. So it starts off with a group from the population becoming separated from the original, and that leads to geographic isolation, which means there's no gene flow. Now gene flow usually allows the 2 populations to mix so that their gene pools look similar. But if there's no gene pool, there's no gene flow, the gene pools are going to start looking very, very different, because there's no migration between those 2 populations. And if they experience different selection pressures, different mutations, they're going to start evolving in different directions. Eventually they become so different that they won't be able to interbreed to produce viable and fertile offspring if they are eventually brought together. So remember the term geographic isolation, and that leads to no gene flow. Selective breeding is when the humans determine which individuals are good for us and which are not good. And we get the 2 good individuals and we breed them together. So we're producing characteristics that are useful for us. And the differences with natural selection is that it decreases genetic diversity much more quickly, because we're mostly just having one or two individuals breeding, and it also does not favor survival. So think about your chihuahuas. They're very, very cute, but if the chihuahua was released into the wild, it probably wouldn't survive as long as the wolf, which is what it came from, pretty much. So they're useful for us, but they're not these characteristics aren't useful for survival. All right, so getting a little bit into the sections that have been eliminated from the course. I'm very happy about this one being eliminated because that was, I think, the worst part of the course. So the fossil record is a record of all the fossils we have ever found, and it tells us a lot about evolution and what was alive and when. And the reasons there are gaps is because sometimes we just haven't found a fossil yet. Fossilization is also really rare, and fossils may have been destroyed. So a lot of people say, well, how do we know that this evolved into this if there wasn't a fossil in between? So we're looking for transitional fossils. They might not have been even found yet. So that's like your counter argument. When you're dating fossils, this is when we're trying to determine how old they are. So relative dating is when we use the age of known rock layers to determine the age of rock layers that are next to it. So this layer, if it's 500 million years old, we know that the layer above it is going to be less than 500 million years old. And that's the law of superposition. Higher rock layers are younger than lower rock layers. That's the law of superposition. Stratigraphy is a branch of geology that uses the relative position of rocks to determine ages. And they use the law of superposition. And using all these different things, that's called relative dating. So that's an estimate of the age of rocks. We don't know the absolute age. Index fossils are really useful in relative dating because index fossils are fossils that we know the age of for certain. And they're around for a very short period of time. So say they're only around from 45 to 50 million years ago. We know that any fossils around the same layer as that index fossil is going to be around the same age. So if we know that this fossil is 45 million years old, we know that this fossil is going to be 45 million years old. Therefore, this layer is 45 million years old. Therefore, the layer underneath it is going to be older than 45 million years old. So that way you can kind of see that we're starting to relative date using our index fossils. So if we find a fossil in Victoria, we know its age. We know that that same fossil, if found in Western Australia, is going to be the same age because we know that it only lived in that very short time period. All right, so methods of absolute dating. This is how we know the exact age of different fossils. Usually we use radiometric dating, so these are the types of radiometric dating. You need to know mostly about carbon dating. And the thing is with carbon dating, you can only use it if the fossil is less than 50,000 years old. If it's older than that, there's not going to be enough carbon left. And I'll go through how radiometric dating works. And the thing is with carbon dating and electron spin resonance, we can only use organic material. So we can only use organisms that are preserved. So we can use bones, skin, that kind of thing. If it's a mold fossil, for example, so if the original organism still isn't there, we're going to have to use the rock around it. So that's when we use these three. So radiometric dating, how it works is you've got carbon-14, and so there's carbon-14 in an organism, there's carbon-12 in an organism. The carbon-14 will start decaying into nitrogen-14. And it decays at a known rate. So it converts to nitrogen at a known rate. We can look at how much carbon-14 there is, and we can look at how much nitrogen there is, and using that information we can guess how much carbon has been converted to nitrogen. And then using that information we can guess how long it's been in the ground. Because we know that every 5,730 years, half of the carbon is going to be converted to nitrogen. So if we know that there's originally 100% carbon-14, and then now we dig up a fossil and there's 50% carbon-14, we know that the fossil will be 5,730 years old. All right, so these are your types of fossils. A mineralized fossil is when the minerals have replaced all the hard parts. A mold fossil is when the original organism has decayed and it's just left a hole where it used to be in the rock. A cast fossil is when a mold fossil is filled with rock. Trace fossil is indirect evidence, so things like droppings, footprints, that kind of thing, without the actual organism. And a true form fossil, is with the original organism preserved. I think recently they found a puppy in Serbia or something that was 8,000 years old, and that was preserved in ice, so that was a true form fossil. So the steps and conditions in fossilization, we also need to know about this. So it's rapidly covered in sediment, and that it has to be rapid because we can't leave time for the fossil to decay or the organism to decompose. There has to be protection from scavengers. So scavengers can't come along and eat up the organism, otherwise it won't be preserved. No exposure to oxygen, that's got to do with bacteria breaking it down. Decreased rate of composition. So all three of these kind of tie into your decreased rate of decomposition. Must remain for a long time for it to become a fossil, and then eventually minerals will replace the hard parts. So the hard parts are much more likely to be preserved than soft parts because the soft parts decompose much more quickly. So now we're getting into a little bit of evidence for evolution. Developmental biology, we're looking at comparative embryology. So we're looking at the embryos of different organisms. And you can see with all these completely different species, they all look very similar in the early stages of development. And that kind of shows common ancestry. So a common ancestor must have given rise to all these different species because the original the early stages of development haven't changed that much, but once we start developing, we're starting to see differences. So we're saying these early stages of development haven't really changed. That indicates that there must have been a common ancestor that used those early stages as well. With structural morphology, we're looking at homologous structures. So homo means similar, homologous structures. And that's when we're looking at structures that were used in a common ancestor. So a common ancestor used the humerus and the ulna for humans, and then they also used it in these other animals, but you can see that the same bones are there, they've just been formed into different shapes for different functions. So even though the same bones are there, they have different functions now, and that shows common ancestry as well. A transitional fossil is the link between an ancestor and a descendant. So the Archaeopteryx shows characteristics of a bird as well as of the Velociraptor. So it shows the transition from the Velociraptor into the early birds. All right, have a go at this question. I'm just going to get a glass of water. All right, I think we might look at the answer now. Whoops. All right, so the answer was C. So potassium 40 half-life is 1.25 billion years. If the ratio is 1:1, that means half of the potassium has broken down, half is still potassium 40. Therefore, one half-life has elapsed, and that'll be 1.25 billion years. Have a go at question 34. All right, so Question 34. It's most likely that the trilobite fossil is present were of the order Q. Perfect. Question 35. All right. The answer is D. And that's because we're looking at the absolute age of the trilobite fossils. So absolute age means we're either using carbon dating or potassium argon dating. And just because it is 250 million years old, we won't be able to use carbon dating because carbon dating only goes up to 50,000 years old. Okay, looking at question 4, I think someone's asked why is it Q? So most likely between 328 to 359. 3. So if the fossil was between these two ages, it has to be younger than 359, and the only fossil that is around for that long would be Q. So that's why the answer is Q. All right, so determining the relatedness between species. If we want to know how related two species are, we can look at its comparative biochemistry. So that's when we're comparing DNA, and we're also comparing amino acid sequences. So the more differences you have, the least related they are. So if you got lots and lots of similarities, that's going to mean that you've diverged from that species much more recently than if you had a lot of differences. Okay, so you can see here that the human is very similar, more similar to the mouse than it is to the chicken, because it's got fewer differences, only 2. And we can use proteins for that in that same concept. So this is just a slide showing us all the things that were mentioned. So we mentioned the fossil record shows that organisms alive today were not in the past, were not alive in the past, and therefore they must have evolved from the older organisms. Transitional fossils obviously show evidence of evolution because they're showing us transitioning from one to another. Comparative biochemistry shows similarities between different organisms. And also shows that they use the same genetic code. Structural morphology, we're looking at homologous structures, so they came from the same origin. Vestigial structures are structures that were used by our ancestors, so things like the appendix. Were used by our ancestors, not used by us, so it shows that we evolved from them to not have to use it. Comparative embryology shows the similarities in development as well. So with phylogenetic trees, this is how we show relatedness between species. So if you guys could have a look in the chat, if you could answer this question, would the bony fish be more similar to amphibians or cartilaginous fish? So type your answer in the chat. All right, so the answer is amphibians. So we have to get this answer, we have to trace it back and see where the most common recent ancestor would be. So between bony fish and amphibians, the most recent common ancestor is over here. And between bony fish and cartilaginous fish, the most recent common ancestor is here. So you can see that the one between bony and amphibians is much more recent than between bony and cartilaginous. So even though these are two fish, the bony fish are actually more similar to amphibians than they are to cartilaginous fish. All right, so there was a question, is it necessary to be able to draw one of these? I haven't seen a question where you've had to draw one from scratch. The ones that I have seen have been like this. So they'll give you phylogenetic tree and they'll give you like a blank box and you just have to fill it fill in the species like that, but it's been very, very simple so far in any exams that I've seen. But I definitely would say practice drawing these phylogenetic trees. So you can probably look up practice phylogenetic trees. All right, pop quiz. Who was the half-blood prince in Harry Potter? All right, everyone should get this one. It was Severus Snape, because he was a half-blood. All right, so human change over time. Last topic in 4.1. We need to know about characteristics that define primates, hominoids, and hominins. So we have to remember that hominoids are still primates and hominins are still hominoids. It's kind of like this Venn diagram where hominoids are inside the big group of primates. So there's primates like lemurs that are primates, but they're not considered hominoids because hominoids are your apes, hominids are your humans and upright walking ancestors. All right, and they're like a subcategory of the other. So with your primates, we have to know certain characteristics, and these are all in my notes, so I won't go into too much depth with them. We've got stereoscopic vision, a long gestation period. So that's how long the fetus or embryo is in the mother before it's birthed. We've got opposable thumb, etc., etc. The way that I remember these is I picture a lemur in my head and I go from top to bottom. So increased cranial capacity, stereoscopic vision, then you're looking at the arms, opposable thumbs, gestation period, that kind of thing. With your apes, you've got things like gorillas, and the main characteristics are the fact that they don't have a tail, they have a really wide chest, and they have a large cranial capacity. With your hominins, there are a lot of characteristics that would be good to know. Those are in my notes as well. So the main things is just things that help with bipedalism, which is walking on two legs. So you want to look at things like the position of the foramen magnum. That's always a popular one to discuss. With the foramen magnum, that's the hole that allows your spinal cord to come out of your skull. So if that hole is very far to the back, you can imagine that your head would be jutting out. And that makes it really hard. Like this with Homo erectus and with like the gorilla, it's really hard to walk upright because you're going to be leaning forward. Your head's going to be leaning forward. So the more forward your foramen magnum is, the more easily you'll be able to balance on two legs. Okay. You've also got other things like the length of your legs compared to your arms. So you can see in the gibbon, the arms are very long compared to the legs, and that's because they're doing a lot of swinging between trees, whereas humans are walking more on the ground. And there's a lot more characteristics, but those are in my notes as well. You also need to know about the history of the human evolution and how different phylogenetic trees can be actually made from the same information. So different scientists can interpret the same information in different ways. So you can see with these phylogenetic trees, there are subtle differences. And you have to be able to use information and argue a point based on that information. And there won't always be a right answer on the exam. They just want to see how you argue a point with the information that you're given. All right, so these are some important species to remember in the hominin evolutionary tree. So hominin is humans and the upright walking ancestors. With Australopithecus afarensis, they were alive 3 to 4 million years ago, and they were clearly bipedal. So the ones before them were kind of bipedal, but they weren't as clear about it. Then there came Africanus, which is 2 to 3 million years old. Then Habilis, Erectus, Heidelbergensis, Neanderthalensis, and those were Neanderthalensis did actually coexist with Homo sapiens, and we can see some Neanderthal DNA in species in populations of humans, Homo sapiens, in Europe and Asia, and that's because Neanderthals moved out of Africa before Homo sapiens, And then once Homo sapiens left Africa, they interbred with the Neanderthals in Europe and Asia. And so Homo sapiens that reside in Europe and Asia will have Neanderthal DNA, whereas those who are of African descent will not have Neanderthal DNA because they never left Africa and bred with the Neanderthals. All right, so you need to know a bit about their age, just very briefly. Doesn't have to be 100% accurate, and you need to know a little bit about what they're mostly known for. So you can have a screenshot of this, and it's also in my notes. So I'll just give you 5 seconds to screenshot if you want. All right, so we need to know a little bit about Homo denisovans as well. So it's believed that they're more of a theoretical species at this point because there was only, I think, a finger bone found from them, and there's actually a link between Melanesian Homo sapiens and Denisovans, and that shows that they interbred with each other. So this is just an example of how new DNA evidence can show us a lot and change up our phylogenetic trees when we find new evidence. So there's 2 main theories about how humans formed. One's the out of Africa hypothesis, and the other is the multi-regional hypothesis. So out of Africa means Homo sapiens developed in Africa, then left. Multi-regional states that Homo sapiens left Africa, Homo erectus left Africa, and then all different parts of the globe, Homo erectus simultaneously turned into Homo sapiens. So we'll have a go at this question. Oops. All right. So, most people chose C, and when we're looking at fossil remains of individuals, Australopithecus, they would show early evidence that hominins were bipedal. So Australopithecus were not descendants of Homo. Australopithecus actually came before the Homo genus. Representative oldest evidence of primates. Primates have been along for a lot longer than Australopithecus. Represents the earliest examples of hominoid superfamily. Same deal there. So they've been around for longer than Australopithecus. All right, we'll skip these questions because we're running short for time. And now looking at 4.2, so area study 2 in unit 4. A genetic engineer is anyone that uses gene manipulation technology to cut, copy, and separate DNA. So those are the 3 things that we do to DNA. To cut things, we use a restriction enzyme, or also known as an endonuclease, and they cut sequences, specific sequences, in a certain way. So this, some restriction enzyme will always cut, whenever it finds this sequence, it'll cut it in the same way. And an important thing with that is that the restriction site is going to be palindromic, which means it's read the same way forwards as it is backwards. So it goes GAATC, then GAATC. And it always cuts it in the same way. With sticky ends, these are sticky ends because they have overhanging branches, and that means they can form hydrogen bonds with the other sticky end. And complementary sticky ends join together, but when you've got blunt ends, those blunt ends will not join together because they're just shaped like this, and they're not very useful because they don't come together at all because there's no overhanging. DNA ligase comes in and it forms covalent bonds between the strands. So there's already these hydrogen bonds between the sticky ends, but DNA ligase comes along, it forms the covalent bonds, which are very strong, much stronger than the hydrogen bonds. A DNA probe is a little piece of DNA that we can label and we can stick in a solution, so when it binds to a DNA sequence that we're interested in, we can know that it's bound to that sequence. Taq polymerase is a type of DNA polymerase. which was found in bacteria that lived in very high temperatures. So Taq polymerase actually works at high temperatures. Human DNA polymerase will actually denature when it gets to around 50 degrees or whatever it is for humans. That's why we use Taq polymerase in PCR, because PCR we're using very high temperatures. It's not going to be very useful if we're using human DNA polymerase and it just denatures. So with PCR, we've got 3 main steps. And a 4th step, which is repeat. So in your first step, denaturation, we increase to around 95 degrees, and you do need to know the approximate temperatures. So around 95 degrees, your DNA strands will separate. So we've got enough energy that those hydrogen bonds will break. And then we cool it down to around 68 degrees, and that's the point at which your DNA probes will bind. And then elongation 72 degrees, your Taq polymerase will bind to the probes and start creating a strand in a 5' to 3' direction. And then we repeat that, so it doubles every time we go through the entire cycle. Gel electrophoresis is a way of separating DNA based on length. So we stick some DNA in these wells and we see how far they travel. So the thing that makes them travel is that there's a negative electrode there and a positive electrode on the other end. Now DNA is slightly negatively charged, so it'll move away from the negative end and towards the positive end. Those strands that are longer will have more friction, so they move more slowly than the smaller strands. So they move through the agar gel way more slowly, and so the strands that haven't moved very far will be called, will be the longest strands, and those that have moved a lot will be the shortest strands. So this is a gel electrophoresis, and you can see we kind of dyed the DNA so we know where they are. The positive test will have strands of known length, so we can compare them, and that's also what this is. This is the DNA ladder. So all those strands will have known lengths, so we can say, okay, this is 300 base pairs. That means anything that has traveled this far will be 300 base pairs as well. The negative control is where we want nothing to happen, just so we're making sure that there's no contamination of the sample. So if we do get little bands in here, we know that some DNA has entered our mixture that we don't want. So some definitions: a plasmid is a circular piece of DNA. It is double-stranded. A recombinant plasmid is a plasmid in which a gene has been inserted. Transformed bacteria is a bacteria with foreign DNA in it. Gene cloning is where we're making many copies of a gene of interest, and we can use bacteria for that, as well as PCR. So with gene cloning, we're using some enzymes. So we start off with a foreign piece of DNA. So that could be some DNA containing a human insulin gene. And we have a bacterial plasmid. So we can use an enzyme known as restriction enzymes, which we've covered, to cut up this the restriction site. So we've got 2 sticky ends here. And we use the same restriction enzyme to cut up the insulin gene. And with the reason we use the same enzyme is so that the sticky ends will be complementary to each other. Because remember, we're always producing the same sticky ends from the same restriction enzyme. So then we stick those, the insulin gene and the plasmid together, and we stick them in a bacteria. Now the way that we put them in the bacteria is either through electroporation, and electroporation creates pores in the plasma membrane, so that is like little holes that the DNA can slip through, or we can use heat shock. And heat shock is where we heat up the bacteria so that the plasma membrane becomes more fluid and the DNA can just slip through. Alright, so once the bacteria take up the plasmid, we can identify which bacteria have the plasmid that we want by their color. So, before we do that actually, we'll have a look at this ampicillin resistance gene. So before we did all these things, we stuck an ampicillin resistance gene on our plasmid. And that's going to make the bacteria resistant to ampicillin, which is an antibiotic. So any bacteria that has taken out the plasmid will not die when we give it the antibiotic ampicillin. And that's how we can select for the bacteria that have taken up ampicillin. The blue colonies have plasmids that have not been, that do not have the insulin gene. And that's because this LAC-Z gene has been cut when we inserted the insulin gene. So when the insulin gene is inserted, it's inserted in the middle of the lac gene. And so the lac-Z gene will turn blue, the bacteria will turn blue if the lac-Z gene is not present. And this is in my notes, so don't worry too much if you don't understand that right now. The white colonies will have a, will not have a lac-Z gene present. Sorry, did I say blue colonies do? So the blue colonies have lactose, do not have the lac-Z gene. They do have the lac-Z gene intact, therefore lac-Z gene breaks down lactose. The white colonies have a lac-Z gene that's disrupted. So the lac-Z gene will not work, therefore it turns white. Okay, and that's just an explanation of all those. You can screenshot that if you want. All right, so we'll have a go at this question 34 first. And we might give like 40 seconds for this one. All right. Most people chose C. So isolate the human insulin gene, then devise the nucleotide sequence. That makes sense. And then V, make artificial gene by adding bases in a sequence gene synthesizer. That makes sense. So we're making a lot of that insulin gene. And by this point, we've actually eliminated all the other answers. So if you're running out of time, you can actually use that method. So you have a look at the first one, and you look at the difference between V and R, and you can eliminate V because R obviously comes before V. Then you can look at the next one between Q and V. We can eliminate V because Q is going to come after R. So that way, without looking at all the other steps, you can actually find that the answer is C. But I'll go through T and the rest of them. So T, insert artificial gene into the plasmids. That makes sense. P, add recombinant plasmids to bacteria and force them to uptake it. Yep. U, identify successfully transformed bacteria. So that was the thing with the lac-Z gene and the ampicillin. And then S, culture the transformed bacteria and harvest the insulin. So we won't do a poll for 35 and 36. The tool used for joining artificial genes would be DNA ligase. So that's what forms the covalent bonds. 36. Artificial human insulin gene has a different nucleotide sequence from human insulin gene, but it produces the same insulin. This is because the DNA code is redundant. Cool. And that's because a lot of different codons can code for the same amino acid. All right. Biological knowledge and society. This is the last point on the course. Almost done. Got 10 minutes to go. GMOs are organisms that have had their DNA manipulated in some way. Transgenic organisms are GMOs that have foreign DNA. So transgenic is a type of GMO. Social and ethical implications. I think you guys should be able to, because we're sending the slides as well, have a look at the slides as well as my notes for these things. You probably don't need me to explain them. The main difference between genetic engineering and selective breeding is that genetic engineering, you can introduce a new gene from a different species. Selective breeding, you have to work with the genes already in that species. In terms of outbreak, endemic, epidemic, an outbreak is a sudden increase in the prevalence of a disease. So things like the flu, it happens every year. We know how much of the flu will happen, so it's not considered an outbreak. If there's a sudden increase in the flu, that will be considered an outbreak. Antigenic drift, small changes in antigens. Antigenic shift is a sudden change in an antigen. That's usually when two viruses enter the same cell and they start mixing. So you're getting a completely different antigen. Conditions for a pandemic, can have a look at my slides for that. I really want to go through experimental design. So in terms of the design, like some questions, a lot of the time they'll have like design experiment question. So you probably don't need to go through all of this, but it is really important to, actually you do need to cover all of this. So you have to have an independent variable. So the independent variable is the thing that you change purposely. You need to describe the dependent variable, and you need to check that the independent variable is the only thing affecting the dependent variable. And you do that through the formation of a control. So a control is a test where there is no independent variable, and that doesn't happen all the time. So for example, if we're testing the conversion using an enzyme to convert some substance A to substance B, we might have a test where there's just substance A with no enzyme at all. And that way we're seeing that enzyme A, that substance A doesn't spontaneously get converted to substance B without the enzyme. So we're making sure that the independent variable is the only thing that's producing the dependent variable. You also want to have your controlled variables. You want to have a hypothesis in there and use that formula for that. Describe your results. So describe what kind of results would support the hypothesis and which would disprove the hypothesis. So you have to say what results actually mean. And then you also want to have these things which increase your validity, reliability, accuracy, precision. So those are some definitions for those. I won't cover those because I really want to go through the Menti stuff. So we'll have a look at the Mentimeter. And so what's the process like to get into medicine? Was it difficult? Okay, so when I got into medicine, it was quite difficult. A lot of people are disappointed when they don't get it, but there are lots and lots of ways to get into medicine. Monash and Melbourne aren't the only unis in Australia. You can go overseas. You can go to, I think, La Trobe as well. A lot of people go interstate to do medicine, and they love it because they get that like college experience that people get in the US when they go. Most people move out in the US when they go to college. It was definitely difficult, but you realize that when you put in the work and you achieve something like that, it's really worth it. But a lot of people put in the work and they don't get it, and that just comes down to chance. I'm really lucky that I did well in my exams when I was applying for medicine, and it is really luck. So I think it's really important to not get down if you don't get into medicine, because it's not got to do with you, it's got to do with how you're feeling on the day. And the ATAR and the UCAT are really not things that show you how good of a doctor you'll be, because like the consensus is with a lot of people in my course, UCAT, UMAT, kind of BS. The things that determine how good of a doctor you can be are like, really does come down to people's skills, and that's why the interview is so important. The interview, basically it's a multiple mini interview, so you get lots of different situations and you have to answer questions that different people have about those situations, and it's mostly like ethical stuff, social stuff, your personal motivations, that kind of thing. But all this is on the internet. You can try and contact me if you have more questions. You can look me up on Facebook, Ethan Fernandez. But yeah. All right. Is differentiation of the chain? Yes. The R chain is the only thing that differentiates different amino acids from each other. Very good question. Any tips for remembering the equations and steps of photosynthesis and cellular respiration? Okay, so the way that I remembered it is using those diagrams that I showed you. So the photosynthesis diagram is excellent. I'm not patting myself on the back there, but use this diagram. It is really good because I learned visually, and it's much easier for me to memorize these things if I have a visual aid. So use diagrams when remembering those steps. In section A, if you find two answers are similar, how do you eliminate the other one? I wish I had a good answer for this, but I don't. On my exam, there were a few questions where answers looked really similar, and that's why I say it really comes down to a little bit of chance on the day. Because if you end up guessing, that is down to chance. How do you eliminate the other one? I'd look at every single word of that answer. I'm sure everyone will do that and kind of like pick it apart. I wish I had a better answer, but I'm sorry. Okay, as an assessor, is there common errors that, this is for Diane, but I'll see if I can answer this one, that students tend to commonly make in responding to questions in the exams? I'd say probably not listening to those command terms that she was talking about. So list, outline, explain, you need to cover all those things. When you're comparing, you have to mention both sides. That was a big thing when I was studying. Those would be the common answers, and not reading the question properly. Is there any particular topics that most bio students struggle with or get wrong? I'm not too sure. If I had to guess, I'd probably say experimental design, but usually the questions aren't too bad on experimental design. Recently, all the topics are getting harder because, I don't know if Diane mentioned this when I wasn't here, but the VCAA is moving a lot towards more application-based questions. So using the information they give you, you have to come up with your own content pretty much. So like on my exam, they asked about what does sterile mean? And that's not on the study design. So we had to apply the knowledge that we had to create our own kind of theory about what sterile actually means. So not particular topics, every topic is getting a bit harder. But yes. How good for time will you be during the exam? Will I have time in the end to check my answers? I found that when I was practicing, I usually had a lot of time to spare. I won't say exactly how long, but on the actual day, I only ended up with 5 or 10 minutes to check over my answers. And that's got to do with exam pressure and stress. Everyone always has exam stress. Even the people who look like they're absolutely fine and chilling, everyone will have at least a little bit of stress during the exam. And that does slow you down a bit because you'll be double checking your answers a lot. And the thing is, you need to trust yourself. So try and limit how much you double check your answers or how much you go back while you're doing your first run through of the exam. And you should have time in the end to check your answers, but don't stress if you don't. Try and get as much down as you can. And especially in short answer, you want to answer as many questions as you can to try and pick up those marks. And it's okay if you make your answers a little bit shorter. And I think doing practice exams really helps you find a balance between having too much information and just the right amount. How do you structure your your study/date in VCE? To be honest, I didn't have much of a structure. At least if you could help me out this one, what do you reckon? I feel like I just studied what I reckoned would help me the most at that particular time. What do you reckon, Elise?
You went into the structure. I was very, very structured. So I timetabled out my entire swap back period. I had the number of weeks and the number of days, and I split each day into a morning session and an afternoon session. I always left my evenings blank and my weekends blank so that if I missed something or I was too slow getting something done, I didn't feel stressed like I couldn't fit stuff in. And then I worked backwards. So from your exam dates, I put practice exams or things that I wanted to do last. I would put that into my timetable. And then I would have next to me a list of all the tasks or things that I wanted to get through before the exam. And I would kind of work backwards and fill in that timetable and structure my time to make sure that I could get everything done before the exam. And then, that way you wake up in the morning and you don't have to think about what to do and you don't have to worry that you're not balancing your study right. You just look at your timetable and you go, it's my morning session on a Tuesday. I'm doing this today. And you just do it.
Exactly, yeah. A lot of my friends did do that where they looked at their normal class timetable. Then they did their subjects according to that timetable, even if they were in SWOT back. Cool. In glucose, is glucose only able to pass through the plasma membrane via carrier proteins? I think that is the primary way that they do it, but I don't think VCAA is expecting you to know to memorize that. I think they can go through protein channels. I'm not 100% sure on that. But I think primarily it is carrier proteins. What's bio at uni like versus bio at VCE? So in med, we did a tiny bit of bio specific, like VCE level bio at the start of the course. And then we started going into more medical kind of concepts. It's from the people that I've talked to, bio at uni, it really does build on your VCE bio knowledge, but it goes a lot more in depth. So the stuff with the regulatory proteins and all that stuff, I have absolutely no idea, because my friends who did science and then they did bio as a major, they know a lot more than I do about the ins and outs of gene structure and all that. Any tips with dealing with procrastination? To be honest, that's something I continually struggle with. I think the main thing with battling your brain with that one is that I find that my brain's like, oh, I really don't want to start studying now, because if I start, I'm going to have to study for 2 hours straight. I think having just saying to yourself, okay, I'll only study for 20 minutes, then I can have a mini break, then I'll study again for 20 minutes. That way you're kind of making it less daunting for yourself. I think that helps a lot. So things like the Pomodoro technique. So if you haven't looked that up, that's really useful. What do you reckon, Elise?
Sorry, I couldn't unmute myself for a second there. Tips with procrastination. I think sometimes you're tired, just give yourself a break, first of all.
Exactly.
But then also try and keep your devices out of your room, little things like that. Don't work in your bedroom sometimes is better. I would always go sit on my bed, and going to a library can be really good. Well, I mean, you can't really at the moment. But going into a space where there's people judging you for being on your phone, go into your living room where your mum can see what you're doing, and then you'll feel more pressure to do work, I guess. But yeah, take a break if you need a break.
Cool. Okay, we are running out of time, so I'll try and go through these really quickly. Are cofactors still in this year's study design? Yes, they definitely are. Do you do undergrad? I do undergrad at the moment. How many exams did you suggest? It really depends. I did 60, but that's kind of overkill because I didn't have anything else to do with my life. A lot of people only did 10 and they still got a 50 study score. So that was really good. Pretty much do practice exams. Do as many as you can, but make sure you're not neglecting your other subjects. How relevant is the stuff you learn now in bio? It is relevant, but they did have a crash course at the start, which was just a week or two for people who didn't do bio. So it's not, it draws on a little bit of bio, but not, you don't have to memorize all the bio to understand med. How can we access these notes? I think that, Gypsy, do you know how they're going to be sent out?
Yeah, just fill in that evaluation that I linked, and I'll link it again. And there's an option there to fill in your contact details.
Cool. I'll probably end it here, and I'll stick around if people want answers to these questions. But please check out that link that Gypsy sent, because we need to have those evaluations for every subject that you attend. Because it helps a lot with improving the lectures, and we need those evaluations to be able to do them next year again. So please fill out those evaluations, and that way you can also get the recording and the notes for today. So with that, I think if you want to stick around for the answer to the rest of the Q&A, I think that's fine. Otherwise, you're free to go. Enjoy VC, enjoy high school. I know it's not the best year for year 12 to happen, but still try and enjoy it anyway, and good luck with everything. All right, so I might just go through the rest of the Q&A for people who are still...
There was just a question in the chat, Ethan. Someone's asked about receiving the recording. Is it correct that they won't receive the recording for a few days?
Yeah, I think so. Gypsy, do you know more about that?
Yeah.
Yep.
So you won't get the recording straight away.
Yeah, if you haven't received a recording, that means we're just kind of editing everything together. We're trying to compile all these notes. You should be able to access them in the next couple of days.
Cool, thank you. All right, so how we meant to remember everything, was there any strategies you used? Bio is definitely a very content-heavy course. Probably one of the more content-heavy besides like history or something. But I didn't like memorize my notes as much as I did lots of practice, and through lots of practice you realize which answers the VCAA is looking for, and so that way you can kind of memorize the answers that they want, not just the facts, but you memorize the facts that VCAA wants you to write down. So that's one way of kind of streamlining that process. And in terms of strategies, I just remember like, okay, so I need to know about the natural selection question that pops up every year. And then I'll just like write down each of the steps. So I'll just say natural selection, get a notebook, write down each of the steps in natural selection. Transcription, get a notebook, write down the steps in transcription. I hope that helps. There isn't really I found that for me, practice exams were my best strategy for memorizing stuff. What do you reckon, Elise?
I agree. Practice exams are very helpful.
Cool.
And like going through the solutions to the practice exams.
100%.
Make sure you correct your exams. Don't just do exams and then leave them there. Make sure you like mark them properly.
Mm-hmm. Yeah, there's no point in doing a million practice exams without learning what you got wrong. And don't disregard the value of redoing practice exams, because when you do the question again, you kind of realize, oh yeah, I remember that from last time. And that the second time around, it reinforces it even more in your brain. All right, do we consider the linked reaction as part of the Krebs cycle? You will know from the question if they are considering it a different stage. So if they give you like they might give you like a table. If there's 4 sections, you know that the linking reaction and the Krebs cycle will be different. If there's 3 sections, usually that means that the linking reaction and Krebs cycle are the same stage in that context. What's the process like to get into medicine? I answered that one. If the enzyme concentration increased but the substrate concentration remains constant, what would the rate of reaction graph look like? So that would just be like that straight line that I said before, because there's no saturation point with enzyme concentration. So the reaction will happen more quickly, but you'll get the same amount of product. Yeah, and yeah. Is there an app we can use for the Pomodoro study technique? I'd say if you don't want to download an app, you can just use a timer on your phone. I found that was pretty useful. There should be Pomodoro apps. There's also apps like that tree app, I forgot what it's called, where like if you open up the app and it grows a tree, the more time you spend on that app, the tree grows. If you use your phone and you get rid of that app, if you go to a different app, the tree dies. So that's kind of motivation. You don't want to kill the tree, so you're not going to procrastinate on your phone. But definitely look those up. There's heaps and heaps of study apps. Can you provide a concise sample answer to the questions that ask you to describe photosynthesis and cellular respiration? I'm sorry if you already covered this in your notes. Questions that ask you to describe. So there's a lot of different questions that they could ask about photosynthesis and cellular respiration. So I wouldn't be able to do one sample answer, but I'd say just remember the reactants, products of each of those stages, and ATP yield as well, and where they happen. So remember those things and you should be fine. Yeah, and someone said Andrew said Focus To Do is a great Pomodoro app available for Android. How do you suggest we go about the biology exams? Just do practice exams for 10 years ago, or go through content as well? So I think what Diane said as well is that just going through content won't help you as much as doing practice exams. So do as many practice exams as you can, and those practice exams will tell you which content areas you want to focus on. So you do a practice exam, you say, okay, I didn't do that well in immunology, I'll go back to my notes and I'll study a bit more on immunology. Practice exams from 10 years ago, they still do have good questions in there, It's just that a lot of the questions aren't relevant anymore. So I'd say, yeah, you can go back to 10 years ago, but just make sure you're only doing the questions that are still relevant. So I'd probably go through the more recent ones first, and then kind of 10 years ago as a last resort. With duplication mutation, is it duplicated onto the same chromosome or another? It's duplicated onto the same chromosome usually. I think if it goes to another one, that'll probably be a mix between a duplication and an insertion mutation. I think that would be very rare. Can you provide a list of the command terms with detailed explanation of what we need to address for that term? I have not created a list with all of those, but I can maybe try and put that onto my notes before Gypsy sends them out. And yeah, hopefully I can do that. I'll try. If I don't, I'm very sorry. You should be able to look that up on Google and look up like VCAA command terms. And that should be helpful. All right, I think that's it. So thank you guys so much for the people who stayed. Am I allowed to give out my email for people if they've got questions?
Ethan, it's Jane here from Skyline. Thank you so much
Jane.
for doing such a fabulous session.
No worries.
People can make contact with us if they want to make inquiry, and we can follow up with you. We're really grateful for your support.
Thanks so much, Jane. Yeah, so good luck everyone with everything. And I hope my notes aren't too confusing. And yeah, have fun.
Thanks so much, Ethan. Fabulous work.
Thank you.